Good morning, all, and welcome to our focus session today. My name is Amrita Jaishankar, and I'm the Executive Director of the Maryland Stem Cell Research Fund. Our goal is to accelerate stem cell-based research, commercialization, and cures. Throughout the day today, you're going to be hearing from experts on the latest breakthroughs and advances in stem cell disease modeling and tissue engineering approaches, as well as gain insights on creating, manufacturing, and delivering regenerative medicine therapies to patients with unmet medical needs. You'll hear more from me in other sessions, but I want to kick things off today by welcoming our speakers and by introducing Dr. Kurt Sibin, who really needs no introductions. He serves as the Distinguished Professor in Regenerative Medicine at the University of Maryland School of Medicine, also as the Associate Dean for Research and Director for the Center for Stem Cell Biology and Regenerative Medicine at UM. So with that, Kurt, I hand it off to you and welcome all the speakers.
Thank you very much, Amrita. This is neat that ISSCR and MSCRF are have put together this session to focus on stem cell research in Maryland, and I'm honored to introduce the other speakers. I'm going to begin with a very short description of a new tool that we're in the process of creating in the lab, and you may find that handy. So let me, as we are finding it or hoping to find it, and so let me share my screen. What we're trying to do with this new tool is to simplify our CRISPR editing of hard to edit cells, hard to transduce and transfect cells, such as hematopoietic and other stem progenitor cells. And as we all know, Cas9-mediated genomic knockout works by Cas9 binding to genomic DNA via a co-delivered sgRNA that's complementary to the chosen target region. Then Cas9 makes a double-stranded scissors-like cut nearby, followed usually by imperfect enzymatic repair, again, most commonly by NHEJ, and the frame-shifting indels, which result from the host repair machinery, end up disrupting production of a functional protein. Now, to do that, the sgRNA and the Cas9 protein must be at relatively high concentration and together at the genomic target in the cell nucleus. So you got to get them into the cell and get them into the cell nucleus. There are many non-viral methods to co-deliver the targeting sgRNA and the large Cas9 nuclease to the cells. The first that anybody uses is plasmid delivery. And that's great for a lot of cells, but not so great for primary cells, stem cells, the leukemia cells that I work with also. So it's inefficient, and it's inefficient in part because it's so toxic. So rather than transfecting plasmids, you can transfect or otherwise introduce RNA, which seems to be less toxic to the cells, but this is still inefficient, kind of a pain in the neck, and the RNA is short-lived. A wonderful way to, when you really have it optimized, is to use a Cas9 sgRNA ribonuclear protein. And this has been great even for gene therapy, where you really need to edit a high percent of hematopoietic stem cells for the gene therapy to have an effect. But it's really laborious to optimize for every single cell type that you want to work with the electroporation conditions. There are several other ways that are out there that I'll just summarize as works in progress at this stage and give you this reference. So as we look through these and try to replace the ribonuclear protein approach that we were using with something simpler, we of course look to the lentivirus as the classic means to efficiently express constructs in hematopoietic stem cells, other tough to transduce cell sites. cell types. And there are two vector approaches to do this. We can take the Cas9 and your sgRNA on in two different constructs and make two different lentiviruses, and you can tag one of them with a GFP and puromycin. But if you get this into the cells and sort the cells based on the GFP, you're not sure that you've gotten the sgRNA into those same cells also. And again, it's two vectors to pair together and optimize. So we then looked at putting these together. So now putting together the GFP and the Cas9 and the sgRNA on the same construct, or alternatively putting puromycin in there for selection of cells if we wanted to do that. And then we said, well, you know, this is going to be really tough, and in certain types of cells, we'd really like a generic way to do this across a lot of cell types. Let's put the sgRNA, the Cas9, and both the GFP and the puro resistance factor together in a single lentivirus. And this is a diagram of the lentivirus we started with, priming and coding in two directions. And here you have off the U6 promoter, the sgRNA, and off a very strong EF1 alpha promoter, Cas9, and also off a similarly constitutive and very strong ubiquitin promoter, the GFP, and the puro separated by an element which leads to their cleavage as proteins are encoded. So we have a few results with this simple first try. And so we put together this complex vector, which requires a bit of engineering to put it together, but once you've got it made, it's all done. And so we put that into cells as either an empty vector or as a knockout vector to knock out a gene called 6-1, where we already knew we had great sgRNA. And we put that in, and you can see that the cells are transfected at about 35%. And then if you culture them for a couple of days further in puromycin, you can get them up to 100%. And you can see that you put Cas9 in both the empty vector and the knockout, and you've knocked down your 6-1. But this really isn't optimal. And while I present the percent positive cells, I don't give you the cell yields, which is not so high for these. So the problem, of course, is likely to be the size of the material in the lenti-vector. And this just gives some more data. I'll skip that. And so we tried to play with it a little, and we put it into an optimized lenti-vector. The sequences out here had been really trimmed down by us previously that we used for a lot of lentiviral transduction. And here we were getting better cell yields, good introduction of Cas9, and we could introduce either a lot or a little of Cas9, depending on how much virus we put in, and we were getting 97% GFP and better cell yield. So optimally, we'd like a construct that between the LTRs is no more than 10 kilobases, and ideally smaller than that. The smaller, the better. So we've been looking to what we can do. Can we make the marker any smaller? Yeah, there's a fluorescent, red fluorescent protein, which you can do called nano, that is about half the size of GFP. If we want to go to different algorithms for design for the PAM sequence, we could use a different Cas9, which is considerably smaller. We can use blastocidin instead of puromycin and shrink the construct a little more. We could play with an EF1 core promoter and see if this does as well as the full size EF1 promoter, and maybe we could play around with ubiquitin and take out some unnecessary pieces from that. So we're going to be seeing what we have to do and how that works to get efficient and easy transduction of a range of cells. We're going to be using leukemia cells and hematopoietic stem cells as our model, but I think in a couple of weeks we'll have something that is ready for others to utilize and further play with. So if you're looking for modify your cells and you are having difficulty, this may be a tool for you in a couple of months. So I wanted to stop there because of we're on Zoom and recorded. Questions are going to be difficult. So I'm going to ask you, for me and for all the speakers, if you can look up our email addresses online and email us any questions. We've all pledged to be glad to answer those for you. So I'm finished. I thank you very much. And I thank you for coming to see this focus session on stem cell research in Maryland. And I'm ready to introduce the rest of the speakers. Our next talk is going to be by Stuart Martin. Stuart is professor of pharmacology and physiology in the Greenbaum Cancer Center at the University of Maryland School of Medicine. And he's going to talk today on microfluidic cell tethering using a tether chip to improve analysis of stem cell sphere formation. Stuart, all yours.
All right, Kurt, thank you very much for the introduction, and hello, everyone. I'm really very happy to be here at ISSCR for the first time presenting this work, which was started together with the Maryland Stem Cell Research Fund and has been developed over the years towards applications and potential commercialization. And so as Kurt mentioned, I'll be telling you about a technology called the tether chip that we've developed to improve analysis of stem cell sphere formation, particularly toward applications in cancer metastasis. And so my lab works on breast cancer, and like many cancers, the limits of clinical detection really prevent us from fully appreciating the process of metastasis. And so in breast cancer, like many other cancers, the patient doesn't die from the primary tumor, which can be relatively easily removed with surgery. It's rather the metastatic recurrence when the cancer spreads to other organs like lung, bone, or brain that really lead to the death of the patients. Now, the problem we have in the clinic is the detection of this metastatic recurrence, because even our most sensitive detection methods like MRI or PET-CT can only detect tumors once they've reached a focal size of more than 10 million tumor cells. And the practical limit is really closer to 100 million tumor cells. So we're only seeing cancer at the beginning and at a very late stage in metastasis when the disease is often too far along to treat effectively. Now, we've known for many years that even small tumors can shed circulating tumor cells or CTCs in the bloodstream at a very early stage. These tumor cells are too large to fit through capillaries, so they rapidly seed distant tissues. Some of these are crushed as they're pushed through the capillaries by blood pressure, but some of these cells escape the bloodstream and persist dormantly in the distant tissue. They go dormant because the distant tissue has a different environment than where the primary tumor arose. But these dormant cells, if they have increased stem cell characteristics, can serve as the seeds of metastatic outgrowth that are going to eventually cause the patient to die. And so these stages of seeding and dormancy really exist below our clinical detection threshold, and we don't have drugs that currently treat these stages of the disease very well at all. So while these stages are invisible in human patients at the current time, we can track them in the laboratory by using tumor cells that express firefly luciferase, making the tumor cells glow. And so if we inject those tumor cells into the bloodstream of mice, within minutes, we can see that the tumor cells trap within the capillaries of the lungs of those mice. Many of these cells are lost over the first few days, but then cells that have increased stem cell characteristics can recur and grow out in the lungs of the mice to form metastatic disease that will cause death in this case. So like many other labs, we're interested in learning how to eradicate these cells at an earlier stage to delay or eliminate metastatic recurrence. And at University of Maryland, we really approached this from the angle was, is there a way that we can learn to effectively target the metastasis of the circulating tumor cells? And this grew out of an observation that we had when we grew tumor cells in a free-floating environment that many people use for forming stem cell spheres. And what we noticed when we grew those cells in a free-floating state, they form these long extensions on their surface that we'd never really seen before. Now, my background in my research before this at Harvard had focused on the actin cytoskeleton. So we thought these extensions could possibly be actin-based structures known as filopodia and invadopodia. So at Maryland, we did a relatively simple test. We took the cells when they'd been free floating for a short period of time, where these protrusions were very short, and we treated with a drug that inhibits actin polymerization that was known to destroy filopodia and invadopodia. And we made the prediction that if these were filopodia and invadopodia, they would go away. Well, that was a short-lived hypothesis, because when we treated with the actin inhibitors, we actually saw that these structures grew longer and more numerous. When we followed up with immunofluorescence, we could see that they had microtubules along their length, but really only actin at sort of these dense ends. And so these were a structure that hadn't been reported before. We named them microtentacles. The reason we name them microtentacles is they help tumor cells cluster together. So what we're looking at here are two free-floating breast tumor cells that have been labeled on their surface with a red plasma membrane dye. We can see they're forming tentacles, and we can see they're attaching to each other. But in order to appreciate precisely what one cell is doing, we transfected with a membrane-localized green fluorescent protein, the cell on the left. And then using a confocal microscope, we can actually image that cell specifically. And what we can see is it's using its tentacles to encircle the neighboring cell to actually form these clusters of cells. And this has gained a lot of interest in the area of cancer metastasis, because circulating tumor cells that can form these clusters have been shown to have up to 50 times higher metastatic potential, but for reasons that people don't entirely understand at this point. The other type of thing we were able to show with this confocal imaging was that if we used a green labeled tumor cell attaching to red labeled endothelial cells that would make up the blood vessel wall, what we could image was the initial attachment of the tumor cell, and then actually through a 3D reconstruction of the confocal data set, we can turn this over and actually see, if you can see that. And so we could turn this over and actually see that the tumor cell was penetrating through the endothelial layer, and actually the tentacles were finding the junctions between the endothelial cells. Now, what the basic mechanism is of the tentacles is actually mechanically based. So that if we image growing microtubules by taking a picture every 5 seconds, what we can see in a cell that's attached to extracellular matrix, the microtubules are progressing outward toward the cell surface rapidly. But where the cell surface is tense, the microtubules get deflected. Where actin is loose, now the microtubules can extend to a greater extent. And of course, when the cell is in a free-floating environment, like it would be in the bloodstream, now the microtubules can extend freely past the actin cortex, and actually this is the formation of the tentacles is promoted by the difference in mechanical environment in a free-floating state. And so the basic model that supports the microtentacles is microtubules originate at the microtubule organizing center and move outward toward the cell surface. There's a network of actin filaments that lies beneath the plasma membrane that squeezes inward on the cell. And it's this balance of microtubules out and actin in that gives the cell a stable structure. Most microtubules have a tyrosine at their C-terminus, and they're very short-lived. They turn over about every 3 to 5 minutes. Through the removal of this tyrosine, through a carboxypeptidase that remained unidentified for a long period of time, a more stable subset of detyrosinated or glue microtubules is formed. And these are the microtubules that we find specifically in the tentacles. The microtubules that are detyrosinated are more stable, because they co-align with intermediate filament proteins via kinesin motor proteins. And together, those two filaments give the tentacle the strength to extend far from the cell body. Now, the enzyme that added the tyrosine back on here has been known for many years, but this carboxypeptidase that removed the tyrosine was unknown for more than 40 years, but was recently identified in a pair of science papers at the end of 2017. And this enzyme actually promotes the formation of the stable detyrosynated microtubules. It makes a very interesting cancer drug target because the current cancer drugs that target microtubules, of which there are many, indiscriminately destroy all microtubules. The identification of this enzyme provides the opportunity to target a specific subset of microtubules that might be more involved in cancer metastasis and therefore also potentially minimize the side effects on many other tissues that the current microtubule therapies impose. Now, we had also shown that mammary stem cells have increases in these microtentacles in some of our early work that was supported by the Maryland Stem Cell Research Foundation. If we separated human mammary epithelial cells into populations that had increased stem cell markers compared to more differentiated cells, we saw the tentacles were higher in the stem-like populations, and those also formed more of these mammospheres in free-floating culture. Moreover, these stem-like cells attached more rapidly to plates than the non-stem cells. And the stem cells also had markers that we know support tentacles like increased fomentin and increased detyrosynated or glue microtubules. But the challenge remained for how do we actually image these free-floating cells? And this was something that meant that we had very few competitors, but it was a problem we needed to solve. So if we imaged a free-floating cell with confocal microscopy, we could get a slice of the image that showed us the tentacles very clearly. But when we collected a series of images to form an image stack, of course, the cell drifted and we got a very blurred image. And so we needed to solve this problem. We did this together through a collaboration with bioengineers at the University of Maryland College Park, where we developed a nanosurface called a polyelectrolyte multilayer that prevents the cells from attaching, but has a hydrophobic lipid incorporated into its upper surface that binds with the plasma membrane of the cell, but still doesn't allow the cell to form protein-based contacts. And this is done through a charged hydrophobic lipid called DOTAP that actually binds to the plasma membrane, and then through its charged end, interacts with the polyelectrolyte multilayer to actually secure it to the surface. And so Julia Ju in the lab had a publication in Lab on the Chip where she actually optimized this technology. And what I'll tell you about now is how we've done this. We've optimized this technology for the analysis of microtentacles, as well as the formation of stem cell spheres. And we're developing assays to test tumor cell clustering, as well as deformability. So looking first at microtentacles, if we allow the cells to attach to these tether chip surfaces, you're now looking at a confocal image of 5 stacks. And if we look at this over time, you can appreciate that the tentacles continue to move, but the cell remains in the center. If you look carefully, you can see cell debris flowing through the background. It would normally wash the cell out of the field of view, but in this case, the tethers keep it strongly in the center of the field of view. We can now introduce drugs like colchicine that destroy tentacles and see them collapse. But we can also look at cancer drugs like Taxol or paclitaxel that stabilize microtubules and actually increase the number of tentacles and their length. The other thing we were able to do by stabilizing the imaging was actually write a computer program. Eleanor Ori, or Echo Ori from University of Maryland College Park, wrote us a program to trace the perimeter of the cells, which you can see here with the red line. And then she wrote a custom MATLAB program that finds the point of local maximum curvature, which actually ends up being the ends of the tentacles. So the stabilized imaging allows us to also get a computational analysis of the length and number of tentacles, and then we can actually judge drug responses in those cells in real time, in less than an hour. But importantly, the optimization of this tether chip, what it's allowed us also to do is stabilize the fixation of tentacles for their analysis. So the tentacles are very narrow. They only are about 100 nanometers in width. And traditional fixation with formaldehyde leads to a rapid collapse of these tentacles. And that's what you'll see here on the left. A cell that's just immediately attached to the tether chip. If we add formaldehyde, you'll see a brief flash in the movie, and it'll be apparent what happens to the tentacles. They get deflected right away, and they almost immediately begin to collapse. And so what Julia did in her lab on a chip paper was she used a heat cross-linking method to stabilize the nanosurface, which left it reactive to formaldehyde. So if the tentacles are allowed to tether to the surface, when we add formaldehyde now, again, you'll see a flash. And if you look carefully, you might see a little flex of a tentacle here. But basically, the rest of them are preserved and stably fixed to the tether chip surface. What Julia was also able to show in that study was that we could then extend this to many other cancer types. And she looked at colon cancer, lung cancer, and prostate cancer. And many of these tumor cell lines also show tentacles. Collaborators here at University of Maryland have also shown that ovarian cancers extend these tentacles as well. All right, and then in terms of preclinical testing, Kiata Thompson in the lab recently published a way to use the tether chip to actually gauge drug responses against tentacles. And she identified a microtubule inhibitor, vinorelbine, that strongly inhibit tentacles. She tests whether it had a specific effect against metastasis by doing a 24-hour treatment of the drug when the cells were transplanted into the breast tissue. And in this case, that single 24-hour treatment did not lead to a difference in survival from breast cancer. But since she knows that the vinorelbine inhibits tentacles, if she did that same 24-hour treatment at the time when the cells are injected into the bloodstream, she could see that in the vehicle control mice, the cells trap rapidly in the lungs, they're lost, and then they recur at about 3 weeks, and many of the mice are dying by 7 or 8 weeks. But a single 24-hour treatment with vinorelbine reduced the seeding of the circulating cells in the lung and strongly delayed the metastatic recurrence, such that that single 24-hour treatment actually improved median metastatic tumor survival from 8 weeks in the control mice to 30 weeks in the mice that were treated with a single dose of vinorelbine for 24 hours. And so this showed us that the microtentacle analysis can rapidly identify drugs capable of reducing metastasis, where identical treatments would be overlooked if we focused primarily on tumor growth. And this is one of the strengths of understanding the principles of stem cell characteristics in tumor cells, is you're not so interested in the growth of the bulk of the tumor, but the cells that are going to actually form the seeds of the metastatic recurrence are the ones that we want to target. All right, and so we've shown that we can do this with cells isolated from the bloodstream. We can judge decreases in microtentacle number and average microtentacle length with a 1-hour treatment of vinorelbine. And importantly, because we can analyze the single cells, we can actually derive 90% power to detect this drug response with as few as 11 tumor cells, which is approaching feasibility for patient biopsies and CTCs. And so then the other thing that we've applied the tether chip to that is also very applicable here in the stem cell meeting is we've shown that this can improve the ability to allow mammospheres to form on the tether chip. Because the cells cannot attach to the surface, they're held in place and they actually form spheres in place. What we saw as we increased the number of cells is that tether cells actually form spheres more efficiently. And a study where we combined red and green fluorescently labeled cells illustrated why. The traditional low attach conditions that people use for sphere forming is obviously measuring a mixture of at least 2 phenotypes: the ability of the single cells to form spheres and the ability of the cells to cluster together. So that if we put 500 red cells and 500 green cells together in the traditional free floating assay, we got spheres that were made up of 2 colors. But if we allowed the single cells to form spheres on the tether chip, they formed single colored spheres, showing that we were getting an isolation of that single cell into sphere phenotype. And we could also see this if we track cells over time here at day 1 to day 5, the single cells, we could see which ones form spheres and which ones succumb to apoptosis. And so this was published by a graduate student in the lab in Scientific Reports. And another graduate student in the lab showed that if we just tracked the cell number at 24 hours, those cells that divided to 2 or 3 cells were highly probable to form large spheres at 7 days. So we could accelerate this assay. And then together with bioengineering at College Park, we're beginning to print arrays of these tethers. So the tethers here are labeled in red, and then the single cells are labeled in green, so we can actually get patterning on these arrays. And this sets up our ability to actually use the tether chip to measure a number of stem cell characteristics that are relevant to cancer metastasis. If we take dissociated tumor cells from either patient-derived xenografts, needle biopsies, or blood samples, we can use as few as 20 cells in a 30 microliter volume to measure microtentacles within 1 hour on the tether chip. If we continue to follow this out to 12 hours, we can begin to see cells dying by apoptosis. And then by 24 hours, we can judge which cells are forming pre-spheres that by 7 days will form into large spheres. And our recent publication shows that we can predict with 97% accuracy which ones will form spheres at 7 days by looking at their growth at 24 hours. And then at the same time, we're developing assays now that require a few more cells, about 1,000 cells, to gauge clustering. But the bottom line here is for just these examples of microtentacles, apoptosis, and spheres, we can stratify the cells from a patient biopsy in different ways, such as those that had high microtentacles, did not have apoptosis and formed spheres aggressively, would be those that would pose a high metastatic risk, where the other end of the spectrum, low microtentacles, high apoptosis, low spheres, would be low metastatic risk. And so then just in closing, what we're doing here is we're trying to accelerate some of the tests that are used for patient tumor cells now. So the current gold standard for measuring tumor cell phenotypes in breast cancer and many other cancers is a patient-derived xenograft, where the freshly isolated patient cells are transplanted directly into mice. In breast cancer, this is known to retain the molecular characteristics of the patient's tumor cell, and it in fact mimics the patient's metastatic pattern. The disadvantages are the PDX growth and metastasis can often take more than 6 months. And so that's very slow to integrate into patient therapy. What we're trying to do is use the live patient circulating tumor cells or biopsy cells immediately. And what we've talked about here today are 3 rapid functional phenotypes of metastatic potential that we can measure in less than 24 hours with the microtentacles, the ability of the cells to undergo apoptosis, and then sphere formation, which is typically taken 7 to 14 days, we're able to accelerate to 24 hours because we can track each cell individually in very small numbers of cells and much more rapidly than a traditional sphere forming assay. And then we're currently working on tests to improve our ability to measure cell clustering. But each of these together within 24 hours can indicate which phenotypes are indicative of tumorigenesis, metastatic risk, as well as measure drug responses in less than an hour. And so that's where we're headed with this technology. As I noted on some of the slides, a patent was awarded for the tether chip technology just last year in 2021. And in closing, I'd just like to thank the members of my lab who actually conducted all these studies. This is a picture of them here, as well as our clinical collaborators in the Greenbaum Comprehensive Cancer Center and our bioengineering and physics collaborators at University of Maryland College Park. This work was supported by an endowed professorship in the Department of Pharmacology, as well as I mentioned very early on, by the Maryland Stem Cell Research Fund, as well as the Maryland Cigarette Restitution Fund, which supports the Cancer Center, and Maryland TEDCO, which is the Technology Development Corporation, which really helped us move toward commercializing the tether chip to the point now where the patent was awarded, as well as the NIH support and support from the Department of Veterans Affairs. So thank you very much. As Kurt mentioned before, please do feel free to reach out with email questions since this is a recorded session, and we'll be happy to address those. So thank you very much for inviting me to participate.
Stuart, thank you for an excellent talk. That's really interesting, stem cell and cancer biology with obvious translational implications. So I'll bet you'll get a lot of emails.
Thanks a lot.
Now, today, if you Google the University of Maryland School of Medicine or University of Maryland Medical Center, you'll probably get first pig to human cardiac transplant, xenotransplantation. So despite the fact that that this is not really, might not really be classified as stem cell research, research on transplantation and the immunobiology of transplant rejection certainly gets very, very close, if not all the way there. So we took the liberty of inviting Mohammed Mahoudin, who is professor of surgery and director of the Cardiac Xenotransplantation Center at the University of Maryland School of Medicine, to tell us about all the excitement in his studies of cardiac transplant that have happened over the years and in the past several months. You're on, Mohammed.
Thank you very much. Thank you, Dr. Sivan, for inviting me, and thank you for the society for having me. I mean, I will restrict my my presentation to the basic science that led to this transplantation, instead of describing the patient and his prognosis and other stuff that happened with the patient. So I would like to start by thanking this patient, Mr. Bennett, who had the courage to participate in this study, and donated his life for us to study this critical critical solution to the organ shortage. So I mean, if you guys have been around and have heard about xenotransplantation, this sarcastic remark has always been around, that xenotransplantation is the future and always will be. But what we have done is, like, we have changed this to what we call now is xenotransplantation is present and always will be. So the why we need xenotransplantation, as shown in this cartoon, there are about 150,000 people waiting for heart or other organ transplantation. Unfortunately, most of them will die waiting for it because there's not enough organ available. And even if all of us become organ donor, still there will be a disparity, and which we'll never be able to overcome. So for that reason, xenotransplantation has been suggested as an alternate for human organ transplantation. This process is not new. The first xenotransplantation was tried in early 1600s, and where a nobleman got some skull defect, which was repaired by a dog skull. Somehow this was rejected over a period of time, and wasn't successful. Then in late 1800s, frog skin was used to repair a burn victim during wars. But one of the most famous xenotransplantation, some of you may remember, is the babyface heart that was done in 1984 in Loma Linda University by Leonard Belly. This patient received a heart from baboon, but lived for only 2 weeks. Other organs were tried, like kidneys and livers, but none of them lived for long. All these transplants were done by using non-human primates, like chimpanzee or baboon. However, over a period of time, after selecting different organs as a replacement, we came to selection of pig, because there are now we know the genome of the pig, and we know how to genetically modify it. They are anatomically similar. They are farm animals, so they breed well in captivity. They have a quick breeding cycle, so they attain the size within months. So baboon for a human transplant will take years to get to the size where its organs can be used, whereas in pigs, it takes only a year. So in Mr. Bennett, we used the heart from a 1-year-old pig. They are consumed in food, so we thought that there would be less ethical issues. And their physiology, as we have learned, that it matches the human physiology. So what happens when you take a pig heart, which is unmodified, and put it in the abdomen of a baboon? This model is used as called a heterotopic transplant, just to study, rejection and have other ways of overcoming rejection. So within minutes, the heart turns into like this, a black block of meat with engorged vessels, all these clotted thrombosed vessels. So in the histology, if you can see here, it shows that that it goes through microvascular thrombosis, and then the myocytes supplied by these vessels die, and with the endothelial damage, the blood enters the intravascular space as shown here. So why does this happen? So all of us, including all old world monkeys and apes, millions of years ago have deactivated one of the enzymes called alpha-1,3 galactose transferase, and because of that, we don't have a terminal galactose on the surface of this carbohydrate chain. Whereas all the other animals shown here, like kangaroo, pigs, and the New World monkeys, have this. So humans are exposed to this carbohydrate in forms of viruses and bacteria throughout our lives. So we produce a large amount of antibodies against this antigen, and it's, as you can see here, the circulating antibody, when it sees the pig vasculature, which has this antigen, the antibody binds to it, and with a complement help, it activates the endothelial cells, which swells up and exposing lamina propria here, and causing the platelet aggregation, leading to thrombosis. And this happens within minutes in front of your eyes. So over years, we have tried to overcome these issues, but I give the example of like onion. We keep peeling off one layer, and we keep finding another issue. We have found at least 3 carbohydrate antigens that are antigenic to humans, and we have antibodies against them or produce antibodies in response to them. And then there are complement regulation pathway incompatibilities between pigs and humans, or pigs and baboons, or there are coagulation regulation incompatibilities between the 2 species. There are several ways there. These were tried to overcome them, and as you can see here, we used methods to block the complements, overexpress human genes in the pigs, and use different drugs to block B and T cell response, and also knockouts these antigens that are expressed on pigs and not in humans. I'll go into more details a little bit later. So historically, this will show you that how far we have come. Initially, the survival used to be in minutes, then it became the survival prolonged to days, then weeks, but then it's kind of stalled until 2005, when until then the longest survival was about 178 days. So we in our group, when I was at NIH, we produced the first breakthrough by using a co-stimulation blockade antibody against anti-CD40 ligand. And with the help of that and pre-depleting B cells, we were able to get the survival up to 238 days. But that was not enough. And also, in this case, anti-CD40 ligand or anti-CD154 antibody found to be very thrombogenic, and a lot of these baboons had thromboembolic complications, and the same kind of complication was seen in humans when this antibody was tried, and the trial was stopped. So next, we switched to another antibody. On the other side of this co-stimulation pathway, we found this anti-CD40 antibody, but when we used it at the same amount, we didn't get any good survival, and that over 238 days was still the longest survival. However, when we increased the dose of that antibody to double the dose, we got a very long survival up to 3 years. So, and this was considered, one of the major breakthroughs by the transplant field. So this is our typical immunosuppressive regimen, which we have used, and similar regimen was used in Mr. Bennett. That our induction requires depleting T cells with the thymocyte globulin and B cells with rituxan, and also complement depletion with the C1 esterase inhibitors or cobra venom factors. We maintain our baboons, in this case with on anti-CD40, a weekly dose, and mycophenolate mofetil, and tapering dose of steroids. In some animals, we have given some anti-inflammatory drugs like etanercept and tocilizumab. So this is the mainstay, and this is the main pathway that we have tried to block. This prevents the crosstalk between the T cells and B cells, and also the APCs and B cells, and prevents activation of B cells and producing the antibody that results in rejection. So this is just to show that the binding is similar in both humans and baboons, but however, this antibody is not is a primatized antibody which cannot be used in humans. So we use a different antibody in Mr. Bennett. This just to show that this antibody it's a non-depleting antibody that can stop the proliferation of B cells and also production of antibody. These are the activation. CD86 is the activation marker. As you can see, that this antibody, the 2C10, which we have used mostly in our baboons, can block the B cell proliferation. So as a proof of principle, what we have shown that that we can stop xenograft rejection over a period of time, over a long period, up to like 3 years. However, when we start removing this anti-CD40 antibody, all these grafts starts to reject. This is the transplant done in the abdomen, which lasted almost 3 years. And this is the last biopsy just before we stopped giving the antibody, and it shows no signs of rejection. And finally, this is the heart, at the time of biopsy. So as a proof of principle, we've shown that that we can get consistent survival for 1 year. Our longest survival was 945 days, and once we remove this antibody, it rejected. So when we remove the antibody, that's shown in 2 animals at those 2 red marks, the non-anti-pig antibody starts to go up. And this causes the rejection, and this in these 2 animals that coincides with the washout of this antibody from the recipient, as shown here in these 2 graphs. There we try to rescue the animal with by giving more antibody, but the rejection was found to be irreversible. So here you can see that this the heart, when we stop the antibody, and in 2 months still beating, and after 2 months it's kind of the contraction stopped. And the most interesting thing was that even after 3 years, the majority of these grafts rejected by the method of antibody-mediated rejection. As you can see, the typical pattern that I showed you in the earlier slides, you see the hemorrhage, coagulative necrosis, and microvascular thrombosis, which has now become the hallmark of the xenograft rejection. So the next logical step we had was to transition to the orthotopic transplantation, which means to transplant the heart in its original place and to see if it's able to sustain life. We have already shown that we can avoid rejection. We have a regimen that can help. So, but all our earlier grafts, failed within 48 hours, as shown in this board that is in my conference room. I just kept it to remind myself that all these grafts failed within 48 hours. And the phenomenon, we describe it as, we call it a perioperative cardiac xenograft dysfunction, where the heart undergoes failure within 48 hours without going through rejection. So the mechanism is still unknown. However, our colleagues in Germany describe a method to overcome it by using this non-ischemic cardiac perfusion in this chamber with a solution that contains a lot of electrolytes and noradrenaline, adrenaline, some hormones, and on top of everything, cocaine in it. We don't know what cocaine is actually doing in the solution, but it's believed that it helps in preventing reuptake of these catecholamines back, so prolongs the effect of it. So with the help of this machine, we take out the heart, we put the heart in this machine for about 2 hours, then transplant in baboons. And since we started doing that, all our grafts have passed that 48-hour mark and have lived for a long period of time. So while we are doing this, we are also with the help of Revivicor, our collaborators, we are trying to modify different genes. As I mentioned earlier, we are taking out a few genes and which are immunogenic to humans or baboons, and also inserting human genes to overcome the graft rejection. And so I'm sure most of you are familiar with how that's done. The pig eggs are taken out, they are enucleated, and then the new genetic information is that we have shown in the earlier slides, where we use CRISPR-Cas9 and other techniques to prepare the construct. That construct is inserted into the eggs. Eggs are injected back into the uterus, and after 140 days, new pigs are born with the new gene construct. There are multiple, gene modifications that have been identified, but which addresses different issues. This slide just give you the example of those issues that we had to deal with. So in this, in our latest model, we are using 3 gene knockouts, which are all carbohydrate knockouts, and all humans have antibodies against these 3 genes, or produce antibodies when they are exposed to these 3 genes. We are putting 3, in fact 2 complement regulatory proteins to help the complement cascade function better. And then we are putting in 2 thromboregulatory proteins to prevent coagulation. And on top of that, we are putting one anti-apoptotic genes to fight apoptosis, and also we are using the human CD47 transgene also to prevent monocyte from damaging the graft. So this is our the final product with 4 gene modification. We have also knocked out the growth hormone receptor because all these grafts were growing big within the pig, within the baboon. So we now are using growth hormone receptor knockout pigs, which control the growths also. This is just to verify that these genes are knocked out or are transgenically present. These are different groups where we have used different combinations of genes, and recently we've published paper where in the life-saving model we were able to survive the graft for almost 9 months. And so that here you can see that again, all those groups and I've shown earlier, we have seen that we have with progressive gene modification, we are able to survive the graft for a longer period of time, and this is one of the longest animals, and surviving for 9 months, which is the longest in the entire world. For the growth problem, we have started to hypothesize that there are like 3 different ways that growth is happening, which is causing graft failure also, and also increasing the size of the graft, and in the chest cavity that becomes an issue. So we think that there are 3 methods of why this growth happens. The intrinsic growth that happened, there's some growth due to rejection, and there are some extrinsic factors like blood pressures and heart rate that makes the heart growth. So we are trying to overcome all these 3 factors also. So as you can see here, this is one of the hearts that had the growth. You can see how thick is the myocardium, whereas this in the bottom panel, this is the heart which lived for a long period of time and you can see that the thickness is maintained as it was when we transplanted. So when you looked at the histology, you can see that one of the factors that caused the growth in this animal was also rejection, because you see typical signs of xenograft rejection with microvascular thrombosis, intravascular hemorrhage, and also vasculitis leading to occlusion of the vessels. Whereas the long-term grafts, they didn't show any signs of rejection. As you can see here, that it shows normal myocardium throughout. So just to summarize what we have done, is that we have successfully translated what we learned in our heterotopic model with the abdominal model to our orthotopic model. This is and we have shown that this genetically modified pig with 10 genes or several other gene modifications, it could sustain life for 9 months. And these animals who lived for 9 months died for some other reasons besides graft rejection. We also shown that we can progressively modify the genes. They may not be enough for human translation, or if needed in human, we have now a process where we can insert or delete further genes. We know that this xenograft rejection cannot be overcome by regular immunosuppression, but we need to block this co-stimulation blockade as shown here. The anti-CD40, as I showed earlier, that was necessary for this. So some other genetic modification that we did in baboons were not very helpful in the baboons, but are useful in the humans. One of the genes that we have removed in baboons exposes another neoantigen that causes antibody formation in the baboon, whereas that antigen is absent in human. So we cannot, we will not have the antibody formation in humans. So there are some limitations of this model, and what we have learned in this model cannot be exactly translated into humans, but the most of the information is applicable to the human. So this was a proof of concept that we took to FDA, and they said to us, okay, no, you need more animals before we let you do a clinical trial. But in the meantime, we had this opportunity. I'll skip this to find this patient who had no other choice left, and with the provision of compassionate care by FDA, we applied for this patient who came to us with non-ischemic cardiomyopathy, with long-standing hypertension, and had already had mitral valve repair. He had several episodes of arrhythmia, ventricular arrhythmias, and required the VA ECMO support. And so we the ejection fraction was only 11%. And due to several factors, and most importantly, his non-compliance, he was not a candidate for an allotransplantation. And so we offered this, the big heart to him, and he accepted, and this heart was transplanted. So this just shows the process that we went through, the FDA, the dialogue, which took about 10 days, and then all the permissions, internal permissions that we had to take place. I just keep this in my office, just to remind us that what we had to go through. And finally, on 7th of January, we did the transplant. I think I'm already over my allotted time, so I'll stop here, and then just wanted to thank the team who took part in this transplant, and also the non-human primate work. And as mentioned, I've just left my emails here for any further questions. Thank you very much.
Mohammed, thank you very much for taking us, as it were, behind the curtain of this fabulous transplant, to all of the elegant work that went behind it, before it, to make the mutations and suppress the immune system to make it possible to do. So we look forward, I think you'll look forward to a lot of emails as well. And I look forward to introducing Valina Dawson. Valina is the Director of Neurodegeneration and Stem Cell Programs in the Institute for Cell Engineering, and she's a professor in the Departments of Neurology, the Saul Snyder Department of Neuroscience and Physiology at Johns Hopkins University School of Medicine. Valina is going to talk to us today on leveraging human brain culture to identify therapeutic targets. Valina, you're on.
Thank you. And I want to thank the Maryland Stem Cell Research Fund for putting this together today, and hoping to share with you several different stories in which we've been using human brain cultures to try and understand human disease. Of course, first let's see. move this forward. First, for the lawyers at Hopkins, our disclosures. We have, in our lab, we study largely stroke and Parkinson's disease. Stroke is the 1 in 6 deaths from cardiovascular disease are due to stroke. It's a major cause of lifelong disability. Worldwide, it is a major cause of loss of life and loss of quality of life. It has always been an important neurologic disorder, but it's taken on a new challenge with COVID-19. The last few months, we've seen papers from around the world describing cases of stroke following COVID-19 infection, for which no other stroke risk factors correlated. So having been infected with COVID-19 is now a new risk factor for stroke, with 1 out of every 3 or 30% of people who've been infected with COVID-19 suffering either stroke or heart attack, often weeks or months after they've recovered from the respiratory symptoms of COVID. So it's becoming even more imperative that we find some treatments for patients who suffer stroke. Right now, the treatment is to relieve the blockage of blood flow, either through enzymes or through surgery, to allow blood to flow back to the brain, and so that these brain regions are no longer injured and have the possibility to recover. But after a few months, recovery stalls, and whatever the patient has as functional as function is what they will have, and they enter into this phase of chronic stroke where not much can be done. We also study Parkinson's disease. Parkinson's disease is the second most common neurodegenerative disease. Again, there are no disease-modifying therapies that slow or halt this disease. There are over 6 million cases worldwide. And the thing with Parkinson's, while people know about the movement part of this disease, the motor control loss, it is actually a chronic global, I misspelled that, neurodegenerative disease that has both non-motor and motor symptoms. And the non-motor symptoms often occur earlier in the disease process, include things like constipation, heart problems, rhythmic problems, autonomic nervous system problems, but also anxiety, depression, hallucinations, and eventually the disease will progress into dementia. And so we don't know how Parkinson's disease starts. There are rare familial cases that have given us clues. From those, we've learned a lot about cellular misfunction that occurs in PD, but in most of the cases that are sporadic, we don't really know how it starts. It's thought there might be environmental components, and there might be risk factor components that combine to start Parkinson's disease. So both these diseases are uniquely human, and we have some proxies in cell models and animal models that have given us some insight, but really what we want to do is be able to study these diseases in human brain tissues and brain cultures to get a better sense of what might be occurring in these human diseases. So the first challenge that we had is that you need to have a culture that expresses both glutamate excitatory neurons and inhibitory neurons. If you don't have both of these types of neurons, you don't have a complete cortical model and a complete proxy. These cultures need to have homeostatic synaptic plasticity and scaling. These are important processes in learning and memory and the normal function of the cortex, and they become disrupted and contribute to disease processes. So we need these. And we also need to have these nNOS neurons. nNOS is the driver for neurotoxicity, both in stroke and neurodegenerative diseases. It also contributes to synaptic plasticity. However, nNOS neurons comprise only 1% of the neurons in the cortex. if you don't have nNOS neurons in your system, you can't observe excitotoxicity or degeneration that's relevant to the human condition or animal condition. And we had shown that in a variety of different models that these neurons were critical to the process. So we took human stem cells and we worked out a differentiation protocol in which we get a proper expression of excitatory and inhibitory neurons. Approximately 80 to 85% excitatory, about 15 to 20% inhibitory, and this would be reflected in a normal human brain. We also have a contingent of glia, because in the human brain, you not only have neurons, but you have astroglia as well. And then when we need to look at microglial contributions, we have to culture those differently and add them into the system. These cultures, when we look at their inhibitory neurons, we're very happy to see that they also were expressed at the levels that you would see in a normal human cortex. So inhibitory neurons, there's a variety of different types, from PV interneurons to the nNOS neurons. nNOS neurons are critically important for neurodegeneration and stroke. The PV interneurons are actually important in Alzheimer's disease. And so if you don't have these in your culture, it's very difficult to study disease in a set of human cells. The next thing that we've looked at in these cultures, and these can be either flat cultures or organotypic cultures, is that they form a physiologic network that behaves in the way that the human brain and an intact cortex would behave. So we look at things to make sure that they have sodium and potassium channels, that they can have evoked action potentials, that we have both excitatory and inhibitory currents, and most importantly, that there's homeostatic scaling in these cells. And this is, you can mimic this by doing a blockade and then releasing it and look at the amplitude of your currents. And in these cultures, we see all of these events, and these are just some recordings that were made in these cultures. So we now have a good proxy for the human cortex that we can start using to study various diseases, cellular signaling mechanisms, and other potential targets, and start to query the system to see if we can find some therapies that might be moved forward for these human conditions. So over time, we have discovered a form of cell death that occurs in the nervous system. It also occurs in peripheral tissues that are acutely stressed, but we're interested in the nervous system, and we've called it parthanatose. We named this form of cell death to distinguish it from apoptosis or autophagy, other forms of cell death that occur and are important in different types of diseases as cancer or heart disease or inflammatory diseases, so that it could be studied specifically and we could find agents that could block this form of cell death. So what happens in stroke, there's excessive calcium influx into cells, which activates the molecule nNOS to form nitric oxide. You end up with peroxynitrite and ultimately activation of this protein poly-ADP ribose polymerase, which generates PAR, or poly-ADP ribose. And that's where the PAR in Thanatos comes from, and Thanatos comes from the Greek god of death. When PARP is activated, then AIF is released from mitochondria. It has PAR bound to it. It picks up this molecule, macrophage migration inhibitory factor, and the complex moves back into the nucleus, where MIF acts as a nuclease, cleaving DNA and causing cell death. So the first question in these cortical cultures was, do they undergo excitotoxic death, stroke in a dish, and parthanatose? And so on these panels here, you can see that very nicely, they undergo excitotoxic death induced by NMDA in a dose-dependent manner. They also undergo cell death due to oxygen glucose deprivation, which would mimic a stroke in these cultures. And they do, the longer the stroke, the more damage they have. And this form of cell death in these cultures is due to parthenoidos. We put in apoptosis inhibitors, necroptosis inhibitors, autophagy inhibitors. They don't protect, but NOS inhibitors and PARP inhibitors do protect, indicating that this pathway is important in human neurons. We were also able in these cultures then to address a sex difference potential between males and females for treatment against excitotoxicity. So males and females, when they have a stroke, will have a different outcome. Females have, particularly before menopause, they have estrogen, and estrogen is protective. So the same stroke will cause a smaller infarct volume in female animals. Because the females have smaller strokes, there had been this thought that perhaps drugs were not as effective in women as they would be in men. And so in this case, we looked at H1 cultures from H1 cells, which are male, and cultures from H9 cells that are female. And these PARP inhibitors, which are all in clinical use for cancer, are all similarly protective, whether it's a male or female culture, indicating that they can protect against stroke and neurodegenerative diseases regardless of sex. And we're starting to see that additionally in animal models. Now, the thing with PARP inhibitors is that not only do they inhibit PARP, but they can trap PARP on the DNA, which makes them very good for cancer treatments, but maybe not so good for neurologic considerations. However, veliparib from Ave is a poor DNA trapper. And so we've chosen to move forward with that in some preclinical IND-enabling studies in the condition for NIH-sponsored SPAN trial, which is a rodent preclinical trial across 7 centers that's being run in a manner that you would run a clinical trial in stroke. And the question there being asked is whether or not we can identify drugs in preclinical models that'll be useful in the human condition. Because the way that you identify a target for stroke and the outcomes that you use are different than the outcomes that are used in the clinical setting. And so hopefully we will find some agents in the SPAN trial that then the agreement is that they will move forward for development for clinical stroke trials. Veliparib is also being considered for Parkinson's disease. Now, interestingly, we took these cells that we had started to, and we put them into culture, but we also took these cells and we put them, injected them into a rodent brain. And this had been done for a different purpose, but we were surprised to find that these human cortical cells integrated into the rodent brain. They survived. They differentiated into different types of cells. And in this case, we're looking at the hippocampus because it's a nice defined region, and it's also part of the learning and memory pathway. And these cells, they develop and form an integrated network with the host brain, which was also a surprise to us. So this offers the opportunity to study human cells in an intact brain in future studies. And possibly in the future, maybe be used for human cell transplants. So now I'm going to shift a little bit or move forward into talking about Parkinson's disease. We know that parthenoidos is very important. It's the end, kind of the way that the cells die, also in Parkinson's disease, as well as in stroke. Different triggers are funneling down. In Parkinson's disease, instead of loss of blood flow triggering it, what triggers Parkinson's disease is misfolding of alpha-synuclein. And so normal synuclein, for reasons we don't know, will start to misfold, and then it will template normal synuclein to continue to misfold until you have kind of this stacking on bricks, and you get these fibrils formed. Eventually, these fibrils become the Lewy bodies and Lewy neurites, which are the hallmark features of Parkinson's disease, and a post-mortem diagnosis is having these abnormalities in the brain. What we think, though, is that this fibril is actually the toxic moiety. The Lewy bodies are not toxic, and maybe they're a survival mechanism, a sequestering of these fibrils into a non-toxic area. Pathologic synuclein underlies a number of neurologic diseases, and these include Parkinson's disease, but these other diseases as well. Now, we now can make these synuclein fibrils in the lab, and we call them preformed fibrils in the lab, or we can take biosamples from human patients, and using a prion technology, an amplification technology, we can get enough fibrils to study. And in doing this, I'm going to just show you the diagram of what we were able to find using both human cultures as well as some of our rodent models, what happens with alpha-synuclein aggregation. So aggregated synuclein gets taken into a recipient cell. We don't know exactly what happens immediately after, but we do know that you get increased nitrosative and oxidative stress. And this permits both the dysfunction of mitochondria, but it also activates this protein c-Abl kinase. And c-Abl kinase is mutated in a human cancer. Gleevec was developed for this, but c-Abl is also expressed throughout the body. And when c-Abl is activated, it phosphorylates both alpha-synuclein and parkin. The phosphorylation of alpha-synuclein promotes its aggregation and misfolding. And we just recently showed that it's a very clear biomarker for Parkinson's disease as detected by mass spectrometry. When parkin is phosphorylated, it gets inhibited and blocked. And parkin is an E3 ligase that mutations in which are a very rare cause of familial Parkinson's disease. So this ties sporadic Parkinson's disease with a familial cause of Parkinson's disease. And we often see evidence for inactive parkin in human postmortem tissue. When parkin is inactivated, AIMP2, which is part of the protein synthesis pathway, moves into the nucleus and it directly activates PARP1 and triggers parthenatose. So that's one of the ways that we end up with parthenatose and Parkinson's disease. It also results in the elevation of a protein we named Paris for parkin interacting substrate. And as an aside, you should never name a death protein after the capital city of France if you ever want to get invited back to give talks in that country. They seem to be a little irritated by that. But Paris, when it's elevated, what it does is it represses the transcription factor PGC1 alpha, which is important to generate proteins that allow for mitochondrial biogenesis and replacement. And so when mitochondria can't be replaced and they're dysfunctional, you end up with a circuit of oxidative stress and enhanced recruitment of these toxic and maladaptive pathways until you end up looking, until the cell ends up dying. We've looked at these pathways both in human cortical cultures as well as our human dopaminergic cultures. And some of the findings that we have are going into the clinic. c-Abl is already in phase 2 and phase 3 clinical trials with new brain penetrant c-Abl inhibitors. And then I'll tell you a little bit about Paris and some of the other pathways. Now, Paris, we wanted to get more evidence that Paris was a good target and could be a good target for therapy. And so what we did is we took iPS cells and we knocked out parkin. We also have patient mutations, but we thought the parkin knockout was a little cleaner and less variable. And then we took these parkin knockout cells and we put a Paris knockdown on them. So the idea is that when parkin is knocked out, Paris goes up and two possible outcomes occur. You have a decrease in biogenesis, but you also have a decrease in mitophagy. And we wanted to tease out and understand whether it was a decrease in biogenesis or mitophagy that was important for cell survival, and whether or not Paris played a key role, not just a complementary or that it was causal, that it wasn't just contributory. So in these cultures, you can see the wild type, we have these beautiful mitochondria. In the Parkin knockout, they're fragmented, they're small, they lose their cristae. This is some quantification of this. And then when we add a Paris knockdown on top of that, we can spare the mitochondria in part from degeneration and loss of function and structure and cristae and of course DNA. And so when we look at mitochondrial DNA in the proteins, you can see this is a Parkin wild type, this is Parkin knockout, and Paris restores this back to normal. So we looked at mitophagy, and this is using a probe, a mitochema probe. And so we have wild type cultures, but the Parkin knockouts have a loss of mitophagy. We knew that this would happen, but the Paris knockdown didn't have any effect on restoring this defect in mitophagy. However, when we put a Paris knockdown on top of a Paris knockout, we can completely restore viability to dopamine neurons. So these are wild type neurons. They die when you knock out Parkin, but when you knock out Paris, their viability is restored. Their mitophagy is not. So we looked at biogenesis using this tag, so we could distinguish between old and new mitochondria. And when you knock out Parkin, we lose biogenesis. But when you knock down Paris, we see an increase in biogenesis. So the conclusion here is that Paris is very important, and the role that it's playing is that it's not in protecting mitophagy, it's in biogenesis. And so if we were able to find a Paris inhibitor, we might be able to restore biogenesis and restore dopaminergic protection in Parkinson's disease models. And this was a very keen controversy in Parkinson's disease, and having these human cultures allowed us to address this specifically in a disease and human relevant manner. And it also then gave us the impetus and to go forward and look for a Paris inhibitor. I'm not going to show this paper is huge, but what we did is a screen for small molecules that would inhibit Paris, and found a molecule, it's farnesyl. We found that Paris is actually farnesylated in vivo, and it's farnesylated, the farnesylation occurs also in human patients. This compound is basically relatively non-toxic, and there is right now development to try and commercialize this finding and do ion and de-enabling studies. And those discussions are underway with tech transfer. The last thing that I want to just present to you, and the way that we use these cultures to understand and develop a new drug for Parkinson's disease, now shifts into astrocytes and microglia and their interaction with neurons. So whenever pathologists look in brains of patients that have Alzheimer's, ALS, Huntington's, stroke, TBI, multiple sclerosis, Parkinson's disease, you always see these reactive astrocytes, and it's kind of a mark for the fact that there are active disease processes going on. People didn't really know why they were there or how they got there. And in a collaboration with the late Ben Barris a couple of years ago, we found that these processes, such as Parkinson's disease and alpha-synuclein misfolded tau, could activate microglia, which then activated astrocytes and caused them to become neurotoxic. So these astrocytes, instead of supporting neuronal function, were chewing up synapses, which are the communication points of neurons, and providing toxic environment, releasing toxins that would kill neurons. So we wanted to know, is there a way that you can prevent microglia from activating astrocytes, or prevent astrocytes from killing neurons? There were some observations in the Parkinson's disease field that never were really totally significant, but were interesting. So there's a compound exenatide, which is used in diabetes to control blood sugar and A1C. And there was an idea that maybe exenatide could also regulate dopamine. And so there was a very small study in Parkinson's disease, which in PD patients were given exenatide. And it seemed that they had a trend towards some improvement in their motor behaviors. Now, exenatide doesn't get into the brain, maybe in homeopathic levels, but Parkinson's patients do have some decrease in blood-brain barrier, so maybe some did get in. And more recently, there was a larger study looking at patients who were on, diabetic patients who are on medication, seemed to have a reduced risk of Parkinson's disease. Now at Hopkins, in neuroradiology, Suki Lee and his father, they decided to take exenatide and they pegylated it with a small peptide. And what they ended up with was an exenatide that had a much longer plasma half-life, it was more stable, and it got into the brain, and it gets into the brain significantly. And so they came and asked us to take a look at NLYL1 and see if it was protective and how it might work. And it turns out that this compound is very protective in animal models of Parkinson's disease and Alzheimer's disease. And so we hypothesized that maybe the way that it was protective was that it was blocking microglial activation of astrocytes and preventing them from becoming neurotoxic. And so in this setting, instead of doing the combined culture as we would normally do, we separated out the microglia and the astrocytes and the neurons. And we put the preformed fibrils of misfolded synuclein on the astrocytes, and they became reactive. And then we take that media and we put it on the astrocytes, and that media activates the astrocytes and they become reactive and toxic. And then when you take that media and put it on the neurons, then the neurons die. And we wanted to know if the compound neurally could block the reactive astrocyte, the conversion to a reactive, microglia, and then prevent the reactive astrocytes, or if neurally acted directly on the neurons. And so in this set of experiments, when we put PFFs on these cultures, the microglia get reactive, and in the presence of the neurally compound, they don't. And so here we're showing that the media from the reactive astrocytes are killing neurons. But if we put the neurally compound first in the microglial compartment, when we get down to the neurons, they don't die. But if you put the neurally compound directly on neurons, it has no effect on whether the PFFs can kill the neurons. But it does prevent human neurons from dying if you do that treatment in the microglial compartment. So this answered this question that the neurally compound was acting on microglia and preventing activation of astrocytes and ultimately neurotoxic cell death. So these are the cellular pathways towards cell death that we've been able to tease out in PD from both animal work, but also our work with human cultures. You have these fibrils that form in a donor cell. They eventually get released. There's transmission. We know this, that occurs between neurons and between cells. LAG3 is one of the proteins that pulls it in to the cell. You get activated stress, activated c-Abl, and then you get this recruitment of this pathway that is involved both in mitochondrial dysfunction as well as activation of parthenoidos. These fibrils also activate microglia, which result in reactive astrocytes, and those also contribute to neuronal cell death. This work and a work of others has led to a number of agents that are moving into various phases of clinical trials past the IND enabling phase. This is really a first in this last, in the last 5 years. We have a number of clinical trials that are based on good scientific studies, knowledge of cellular dysfunction and cellular pathways. And so we're very hopeful that these studies will bear fruit in Parkinson's disease. We've been assisting people with the c-Abl kinase inhibitors. These are in phase 1 and phase 2 trials. The GLP-1 receptor, this is our company Neurally, but also other companies had GLP-1 receptors agonists, and they are also moving in to treatment of Parkinson's disease with these agents. And our companies now are poised to start phase 1b trials in Alzheimer's disease as well. So this is a unique time period. We have, in addition to those compounds that are in phase 1 and phase 2 clinical trials, there are a number of compounds that are in IND enabling studies as well. So there are PINK1 and Parkin activators. We're looking at Paris inhibitors. And we also are looking at MIF inhibitors to prevent the last stages of parthenitos. To a neuroscientist and to neurologists, it's just wonderful to have a cornucopia of potential agents that could be disease modifying and stalling the disease for Parkinson's disease, may slow it down and hopefully block the progression, allowing patients to lead a normal lifespan. This work has been done by a host of fellows and students, and we're grateful to all of them. Also to our collaborators within Hopkins and without and internationally, and of course to the folks that have funded this work. And the human culture work and stem cell work was, it started in collaboration with the Maryland Stem Cell Research Fund, and they've been funding us for a number of years to help develop these models and really push them forward so that we can get good targets that we can take forward into the clinic and in Maryland-based, hopefully, and as well as other pharmaceutical companies. So with that, and thank you very much.
Thank you so much, Valina. Well, it was a great pleasure to participate in this symposium today and to show off some of the science that's happening in Maryland. We've got a lot of different kinds of stem cell science happening here, and we look forward to your questions, perhaps right after the ISSCR meeting. So please send us all emails. I want to thank the speakers personally, not only for their great talks, but also for staying on time. Amrita, it's yours.
Great, thank you. I just want to thank the speakers as well, and thank you all so much for your time and for listening to us. Thank you. Hi all, good morning, and welcome to our focus session at ISSCR, and thank you for joining us today. It is my honor and delight to welcome all of our speakers today. The way this is going to work is that we're each going to introduce ourselves and give a short presentation, and then we'll head into the panel discussion. So I'm going to kick things off quickly by introducing myself and the Maryland Stem Cell Research Fund. So my name is Amrita Jaishankar. I'm the Executive Director of the Maryland Stem Cell Research Fund, or MSCRF. At MSCRF, our goal is to accelerate human stem cell-based research, commercialization, and cures. We've invested over $170 million in over 500 awards across 6 different programs to achieve this mission. So our focus really is on identifying and fostering cutting-edge research and innovation in the field of regenerative medicine in Maryland. We have 6 programs that are broadly structured around what does it take to move a regenerative medicine discovery from the lab where the invention occurred to the clinic where it can reach patients. So we support everything from early stage discovery through commercialization to clinical trials, as well as postdoctoral fellowships for that next generation of scientists and leaders that are going to grow in this field. Our clinical trial programs are also open to any US-based company with a clinical trial site in Maryland. So while we're all about stem cells, we're really disease agnostic. In just the past year, we've supported world-class organizations and companies looking at a diverse, wide range of disease indications, including rare diseases to more prevalent ones like cancer, Alzheimer's, stroke, cardiac disease, which account for the top 10 leading causes of death in Maryland and across the nation. So we're really committed to shaping the regenerative medicine ecosystem here in Maryland broadly through 3 different pillars. So we foster research and innovation, we build great stem cell companies, and we serve as the connective tissue. So through our university-based programs, we encourage innovative, high-risk, high-reward ideas, that traditionally don't get regular funding. And then we are in early, building teams, shaping research, and driving tech transfer and commercialization. Through the second pillar, we support milestone-based programs to create value, to de-risk technologies, and really to advance science and accelerate cures. And then through the third pillar, we really connect the whole regenerative medicine ecosystem. We build various alliances and partnerships. We collaborate with universities and various local and global stakeholders and really serve as a resource for our community. So we're really excited to be here today, and throughout the day, you're going to be hearing from key individuals from each of these pillars, whether there are universities that we partner with, whether it's all the companies that are here on the panel today, or our collaborators, including those at NIH who are on this panel. And of course, we're thankful to ISSCR for this opportunity and glad to be here. Of course, everything starts with great science and innovation, and we're really proud to be partnering with our world-class universities and research institutes, and also proud to be supporting our growing portfolio of companies, many of whom have raised follow-on funding. We've had a couple IPOs, and you can learn more about all of them and about all of our funding opportunities on our website at mscrf.org. You can also learn more about the science that we support and the impact that we've helped create through our YouTube channel. You can also stay in touch with us through our app. And I'm just going to end this by saying we truly are about building a community, committed to giving every person, every project the best chance to succeed. And at the end of the day, that's really personal. And it's personal for many reasons for all of us. For me, it's personal because I trained as a stem cell scientist here in this region, and this is my community. It's personal because we each have or know someone with one of these diseases that we're trying to cure. It's personal because we have this hands-on approach to working with everyone that we fund and even those that we don't to make sure they're successful. But above all, it's personal because of patients like Yatunde and Lily and countless others whose lives are saved by our collective efforts to accelerate stem cell treatments and cures. So I want to thank you for your work and time and leave you with my information if you would like to engage with us. Thank you, and I hand it over to the next speaker, Dr. Sashank Reddy.
Amrita, thank you so much for inviting me to speak today. My name is Sashank Reddy, and I'm a faculty member at Johns Hopkins in the departments of plastic surgery and biomedical engineering. And I'm also the senior medical director for Johns Hopkins Technology Ventures. And in that latter role, I get to interface with a number of the promising stem cell therapeutics companies and gene therapy companies that are coming out of Johns Hopkins. It's a great privilege to be able to collaborate with the Maryland Stem Cell Research Foundation to bring these technologies to patients. It's something I'm very passionate about, and many of us are. Today, I'm going to tell you something that is really at the intersection of those things, which is about a company that I helped start out of Hopkins called LifeSprout, that is using technologies developed in my lab and that of my collaborator, Dr. Haichuan Mao, to solve what we're calling the last mile problem for cell therapies. And I'm deliberately leaving the term stem out of it because it applies to stem cells, but it also applies to the differentiated products of those stem cells. And what I have in mind here is that the regenerative cell therapy field as a whole has enjoyed a lot of success over the last decade in being able to create end cell types of interest. So if you want to make a beta cell for diabetes or a cardiomyocyte or a specific type of CNS neuron or photoreceptor, our capacity to be able to do that from relatively undifferentiated precursors, iPS or ES cells, has gotten very mature. However, our ability to get those cells to survive and functionally integrate in diseased tissues has lagged behind. That's what I call the last mile problem. How do you get these painstakingly created cell therapies to actually do their job in the patients at the locus of disease? That's where we want to play in LifeSprout. We want to use our synthetic biomaterials to be able to support those cells in their function. And that's what I want to tell you a little bit about today. I'm going to skip the executive summary and the team slides here. So the mission for our company is to develop these revolutionary products for tissue replacement and regeneration. The materials with platform that we're going to describe, again, is an outgrowth of work that was done at Johns Hopkins and funded in part by the Maryland Stem Cell Research Fund. And it really centers on this core technology, which we're calling the regenerative matrix. It is a nanofiber hydrogel composite material that both looks, as you'll see here, ultrastructurally, and as you'll have to take my word for it, performs until the next few slides, very much like living tissue. So what I'm showing you here is an electron micrograph of our nanofiber hydrogel composite platform, the regenerative matrix on the left. And what you see on the right is soft tissue. This is just fat from an experimental animal. And the architectural similarity between these two things is readily apparent. You really don't even need to know any science to see that this looks like that. And that's despite the fact that our materials are completely chemically defined and a product of synthetic chemistry. There are no cellular or animal-derived materials that go into what we're doing. These aren't decellularized native tissues. And yet we can get that intense biomimicry. And what's even more special and important in the context of cell delivery is what happens when we implant these materials into animals and then ultimately into our patients, we hope, is that they get vascularized and functionally integrated. So what we're showing you in the upper panel here is the regenerative matrix after it has been implanted, in this case in a rodent model for several months. You can see that it is red. It almost looks like tissue. It's becoming vascular versus a standard hydrogel material like a hyaluronic acid in this case, completely clear and looks synthetic, non-living. On the lower panel, you see the consequences of an indocyanine and green injection into this animal, and that lights up vascularized structures. And again, our materials very well lit up by this, and the hydrogel alone does not, suggesting that the materials remodel and become vascularized to resemble living tissue. We published on this in Science Translational Medicine a few years back. More recently, we've put a paper out in Plastic and Reconstructive Surgery detailing how these materials on their own can encourage natural tissue remodeling, vascularization, and regeneration, and also can enhance the delivery of cells. And we began with a readily abundant cell type, adipose-derived stem cells and adipocytes, which are present in all of our bodies and easily accessible through liposuction procedures. And we have recently used this as the first example, the first manifestation of regenerative matrix enhanced cell delivery. I'm going to skip over this slide, but the important thing to say is that we've already tested these materials now in patients. We have a clinical trial ongoing in Europe where more than 100 patients have been injected with the regenerative matrix materials in its first product manifestation, which is just the materials alone. And we have several programs in the pipeline that we want to extend the utility of this technology into the area of cell delivery, again, to try to address that last mile problem. One we're particularly excited about is what we're calling regenerative cell therapies, LSO2, programs in trauma and in breast reconstruction, quintessential soft tissue defects, where delivering cells like adipocytes and adipose-derived stem cells in a more predictable way could have dramatic impacts for patients. And I'll just close with a little bit of science to illustrate how that might work. So on the left-hand panel here, we're showing you what happens when you just deliver regenerative matrix and lipoaspirates, so just easily accessible adipocytes and ADSCs. The volume drops by about half and then plateaus versus a nearly 80% loss of the material when you just try to deliver cells alone. Going back to my original thesis, that we can make cells, we can deliver cells, but when they're divorced from their healthy environment or a mimetic of that healthy environment, as we're demonstrating here, they don't really do well. They don't really survive. They don't functionally integrate. We've also shown that not only does it improve the volume, it improves the cellular architecture. So the RM-assisted lipotransfer looks essentially indistinguishable from the adipose and soft tissue matrix of the native tissue in the surround. There are multiple viable adipocytes present in our materials, unlike when you do the lipotransfer alone. So we're really encouraged by this and are hoping to start a clinical trial, a pilot trial, again in Europe, examining the use of these materials with cells to address patients with discrete indications. And we have broader aspirations to do that for the large number of patients who have soft tissue defects and potentially beyond those indications as well. I'm just going to stop there. I'm going to thank the Maryland Stem Cell Research Fund, which was a key supporter of ours in the early days of this work and continues to be. And I'll turn it over to my colleagues for the next presentation.
Thank you, Amrita. Really appreciate you and Maryland Stem Cell Fund Research Foundation inviting Vita Therapeutics to be here to join this ISSCR panel today. To formally introduce myself, my name is Douglas Falk. I'm the co-founder and CEO of Vita Therapeutics, which is a cell engineering company that is really focused on combining traditional gene editing with traditional cell therapy, all with the hopes of potentially replacing defective cells and patients with functional cells. So we got started about 2.5 years ago at Johns Hopkins University by myself and my co-founder, Dr. Peter Anderson, who's also our Chief Scientific Officer. And at Vita, we really believe that we have a curative therapy for a number of different localized muscular dystrophies. And this research stems from our neuromuscular platform, which was invented out of Dr. Gab-Seng Lee and Dr. Catherine Wagner's lab at Johns Hopkins. And then we also believe that we have a very effective treatment for multiple different solid tumors. And this came out of Dr. Alan Freeman's lab and is really the core foundation of our immunotherapy platform. So to go one step deeper, we really have 3 core platforms that we work with here at Vita. The first of which is really the only thing that we've been public about to date. As we launched our Series B just a few months ago, we did it with the launch of our second and third platforms. But nonetheless, to introduce you to all 3, the first is the neuromuscular platform, where we're using iPSCs to derive a very specific cell type known as satellite cells. Satellite cells are the very foundation of muscle biology. Once our muscles are injured, a series of mechanisms occurs that essentially starts the process of repairing and replacing human muscle. So that's exactly what we've been able to show preclinically. We can actually repair and replace and regenerate healthy human muscle using our iPSC-derived human satellite cells. So long-term, our goal is to actually treat a number of different healthy or different degenerative muscle conditions, and at the same time move into multiple different muscular dystrophies. Long-term, our goal is to actually use these satellite cells to treat a number of different degenerative muscle conditions, but at the same time, treat a number of different localized muscular dystrophies. So to start, we actually chose a specific disease with a very high unmet medical need, known as limb-girdle muscular dystrophy 2A. And we've put together a preclinical program called VTA100 to actually move that into the clinic. Right now, pretty much every single partnership that we need to get these cells into the clinic has already been fully established. So it all starts with the CDMO, somebody who can take our research protocol, optimize it to that of a GMP protocol so that we can actually use these cells to treat real human beings. About a year ago, we actually partnered with CDMO. We've now done that. We fully optimized our research protocol and created a GMP protocol. So the next step is really moving into the IND enabling cells study process, which we've also got 2 different partners, 2 different CROs that we have contracts in place for, and we plan on initiating these studies a little bit later this year. Recently, we had a really great interaction with the FDA, where we actually presented a lot of the data that we had done historically. And the good news is now, after that meeting, all we have to actually do is replicate the exact same studies that we had done, but instead of using the research grade cell, now use that of a GMP grade cell. And then finally, really the last piece of the puzzle here, and quite frankly, the most humbling piece, is actually the patients themselves. So we went out, we partnered with 3 of the top muscular dystrophy sites in the US for our phase 1/2 clinical trial. And then once we had that in place, we actually went out and started talking to real LGMD2A patients. And what I can share is, as of today, we actually have over 50 plus US-based qualified LGMD2A patients who have already volunteered for our phase 1/2 clinical trial. And we've already actually drawn blood from some of them. And the GMP production is currently underway. So right now, we're anticipating filing that first IND a little bit later into the second half of next year. But really, we chose LGMD2A because we thought it would be the perfect disease to give us that proof of concept that satellite cells can repair and replace human muscle. So after that, our goal is to really start moving to other different types of muscular dystrophies, and then beyond that, different degenerative muscle conditions. Our second platform is actually an immunotherapy platform. So here we actually extract a specific cell type known as myeloid cells. We then have a proprietary process where we take those myeloid cells and we actually genetically lock them into their tumor cytotoxic M1 killer macrophage state. We have some great proof of concept data that suggests that that alone could be a really nice anti-tumor efficacy effect in prostate cancer models, pancreatic carcinoma models, neuroblastoma, and also triple negative breast cancer. And now we're really in the final process of taking that technology and combining it with some next generation CAR technology, which we actually think is going to lead to a more robust anti-tumor efficacy effect. So that CAR technology I referred to is actually something that's very proprietary to Vita that we actually refer to as the carrier technology. And just recently, we actually established some really great proof of concept data that suggests that that alone, without the genetically engineered myeloid cells, can actually have a really robust anti-tumor efficacy treatment, as well as it relates to triple negative breast cancer models. So now we're really in that final phase of development where we're going to marry these two technologies together because we think these genetically engineered myeloid cells that are transduced with our carrier technology is really going to be the ultimate treatment that's going to have the most benefit to patients. So over the next 6 months, we're going to be looking to test this out on various different preclinical models. We hope to have a specific indication identified by about Q3 of this year, at which point we'll start the formal preclinical process. But at this point, we are planning on filing that IND towards the end of the first half of next year as well. So we really consider both the neuromuscular platform and the immunotherapy platform really the co-leading programs that we have here at Vita. And then finally, that leads us to our third platform, which is really the long-term goal that we have. And that's to use iPSC technology to actually derive one universal hypoimmunogenic cell bank. So one cell line that's completely evading the immune system that we could use to treat a whole bunch of different patients, regardless of their DNA, and at the same time, drastically reduce the cost of development. So what we've done is we've actually licensed in a hypoimmunogenic technology and then filed a few other patents around that technology that just include a few other edits that we think are necessary for long-term evasion. And really what we anticipate happening is right around the same time that we actually get phase 1 proof of concept data in our first two platforms, that's when we are going to be looking to move forward with this hypoimmunogenic technology. Right. So what's really unique here, I think, about Vita is we really set the company up where we're focusing on both the autologous approach, but then also this hypoimmunogenic approach. And I think that's really important, really for one simple reason. We want to show and prove that our cell types can actually, work and perform functionally in patients before we then worry about evading the immune system. Right? We want to show that our satellite cells can effectively repair and replace muscle. We want to show that our myeloid cells can effectively treat solid tumors. So what we anticipate happening is that GMP manufacturing for this hypoimmunogenic technology, as well as the IND enabling studies, we're hoping that they kind of wrap up right around the same time as we achieve that phase 1 proof of concept data, so that we can then look to put together a separate clinical trial using that technology. So hopefully that gives you just a really quick, good understanding as to what technologies we have here at Vita. And then lastly, I'll just touch upon the projection and historically what we've done from a financial perspective as well. So we started the company back in 2019. We raised a really small seed round, about $2.5 million, to go along with about $4 million of historical grant money. Really allowed us to just set up the company right here in Baltimore, bring on a number of scientists full time, replicate the science that we had been doing at Hopkins within our own lab, and then really enough resources to put together the big vision that has now become Vita. So early last year, we closed a $32 million Series A. Those funds take us through the end of this year and into the start of next year. It really gets us to the pre-IND meetings for both those two leading programs. And then just a few months ago, we launched a $40 million Series B. Off to a very good start thus far, have about 31 million committed. But what those funds are intended to take us through is actually getting the INDs for both the neuromuscular immunotherapy platform and starting those phase 1 clinical trials. So hopefully that just gives you a really good understanding of who we are and what we're working towards. Thank you again, Amrita and the Maryland Stem Cell Foundation. Really appreciate all the support that you guys have given us over the years. But I'll stop there, and I believe I'll be turning it over to Dr. Barty.
Thank you, Doug. Let me just quickly share my slides. So I want to start by thanking Amrita for this invitation to talk about our work trying to develop a cell therapy for a disease called age-related macular degeneration. So essentially, when an AMD patient goes to the eye doctor, what the doctor sees is on the left side of your screen is a schematic of a back of a AMD patient eyes called the fundus photograph. And the patient has a big lesion in advanced dry AMD stage, this gray area where they're completely blind and they can't see. And that's where our central vision is. So those people are completely blind in the central part of their vision. If you look at the right side, they're blind because this would be the middle part of the eye, which is where they lost all the photoreceptors, which are the light sensing cells. But they've lost these photoreceptors because the underlying support of this cell type called the retinal pigment epithelium, in short, the RPE, is gone. And these cells provide nutritional support to photoreceptors throughout their life. So because RP cells die off, photoreceptors die, hence the patients go blind. And our approach has been to start in the advanced patients and really focus on the edges of the lesion where there are photoreceptors still kind of surviving and they're destined to die because the RP support is slowly going off, is to transplant the RP patch in the what is called the transition zone of this lesion, which you see on the left side. Our transplant is a 2x4 millimeter RP cells on a biodegradable patch. And the idea is that this patch will preserve the dying photoreceptors and stop the disease from progressing further. And eventually, once we demonstrate proof of concept in some advanced patients, we can go to earlier disease stages and really stop the disease from happening at all. So our manufacturing process this is an autologous iPS RP patch. What that means is we start with patients on blood, make their iPS cells and make the RP patch, take it back to the patients. So the whole process takes about 6 months. Needless to say, we have optimized and standardized every single step of the process, including days where how the reprogramming, making a mini bank and a working bank of iPS cells, the QA that has to be set up for iPSC reprogramming and bank qualification, and then leading into the differentiation, again, going over steps into differentiation. All this release criteria has been set as part of the IND enabling studies that we performed, leading up to the final product, which is where we seed progenitor cells onto a scaffold, making a patch that gets released based on its functional characteristics and purity, and of course, sterility that is given for every cell therapy product. So just to quickly show you how our cell therapy product looks like, it's based off of a biodegradable patch, which is made of a material called polylactic glycolic acid. Here you see a scanning EM of the scaffold with nanofibrous electrospun fibers of the scaffold, and they form a mesh so that the cells can stay on top and form a functional and fully mature monolayer. And this is how the scaffold, once the cells are ready, the transplant looks like. This is a scanning EM of our transplant, pseudo-colored to show you the scaffold in blue. And you see cells from a side view, 3 cells here, and the nucleus in brown. And you see these apical structures in green. These are the kind of what I usually call the business end of RP cells. These structures interact with the photoreceptors and help support the photoreceptor health and functionality. And this, what you see, I hope you can appreciate, is a very homogeneous, fully differentiated RP cells. And the highlight of that is that they form these apical processes that what tells us that these cells are fully differentiated. So once the cells are ready, the process was optimized. We performed in the IND enabling studies runs from 3 independent donors as part of our engineering runs and 3 clones per donor, and demonstrated that we can get pure RP cells. So you can see all the cells lack pluripotency markers and have almost 100% committed RP markers, and even mature RP markers are above 80-90% in these cells. And by functional characteristic, which in the interest of time I'm going to skip today, these cells do perform like really native human RPE cells. In animal models, we demonstrated as part of GLP tox studies that if we transplant a 0.5 millimeter patch, which would be the human equivalent 2x4 millimeter dose that I showed you earlier, this patch in an immunocompromised rat fully integrates into the back of the eye. And as you can see here, the red cells are the human cells labeled with the dye, an antibody for antigen called STEM121, First of all, what you see is that after 10 weeks, the scaffold is completely gone. That's what you expect from a biodegradable patch. And the entire patch has integrated itself. All the cells have integrated themselves into the back of the eye to the point that they actually start interacting here with the rat RP cells. And after performing this in about 450 rats as part of the GLP tox studies, we could prove to the FDA that these cells do not form any tumor or are not toxic to these animals. As part of the efficacy studies and feasibility surgical procedure, we use mini pigs as a model because we wanted an eye that is in size and anatomy similar to human eyes. But in mini pigs, unfortunately, there's no model akin to AMD-related degeneration. So what we did was we use a micropulse laser to laser ablate the pig RP cells. And the reason why it works is that if you use a specific wavelength of laser 532 nanometer, it's specifically absorbed by the pigment in RP cells. So the cells heat up and they die. And as a result, the photoreceptors would die, very similar to what would happen in AMD patients. And then we were to transplant RP patch here and protect the overlying pig photoreceptors. So here's a brief data from the pig studies. On the left, you see an infrared image of a laser damaged RPE. So normally the infrared light would not pass through the pig eye, but if we laser ablate the RP cell, the light passes through. We call it a window defect because now the light goes through. It's a 7x4 millimeter window of RP cells that we ablate. By histology, we can prove RP cells are gone. As a result, photoreceptors are starting to die off, and the idea would be to transplant human RP patch and have it protect the pig photoreceptors. We optimize a surgical procedure. I'm going to briefly show you how the surgery is done in a pig eye. The eye has been prepped. Surgeon is making the hole in the eye wall bigger to put the transplant. And you will see in the next shot, the transplant tool inside the eye. And you can see the tool, the retina has already been lifted, cut has been done, and you can see the RP patch with pigmented cells in the tool. And surgeon is aligning the tip of the patch to the retina cut and will deliver the patch under the retina. And retina is just flattened on top of the patch and it just stays in one place, which we can test right after that using a technique called optical coherence tomography or OCT, which looks at reflectance from the back of the eye. And here on the left side of your screen, you can see the patch, just looks like a patch. And this bright spot is a cut in the retina. And if we do OCT section through it, you can see here in the middle panel, the yellow line highlights the RP patch. And right above it, the retina looks fairly healthy. If you were an OCT expert, you could tell the difference in quality of the retina above the patch or further away from the patch. Here, you'll have to take my word for it that the retina on the patch heals faster and at a much faster rate than the retina away from the patch, which has also been laser damaged. But we have another technique that we can use to look at functionality of the retina. This technique is called multifocal ERG because retina is an electrically conductive tissue. You shine light, you can measure electrical response from the retina. Here, what we're doing is, this is a light shining in the same eye on the healthy part further away from this area. We get nice and big response. The red is light shining to the laser part, but no transplant, and we get almost no response. And green is the area where the light is shining on the transplant, and we get a response in between, suggesting that human RP patch is protecting the pig retina from dying and preserving its functional responses. We can confirm this once the animal is euthanized. We can confirm this by histology. Two different areas of the same eye, laser area with no transplant on the left, laser area with the transplant on the right. Here you can see the yellow line highlights the transplant. ONL is autonuclear layer. That's the thickness of photoreceptors, and you can see that it's double in this area as opposed to where the transplant was not delivered. So really, human RP patch protects pig photoreceptors from dying. So this work, the IND was submitted in 2019 and was approved by the FDA for a phase 1/2 clinical trial to test autologous patch in AMD patients. We started the studies despite some delays with COVID. We have now started the clinical trial at NIH. We have 2 cohorts of advanced AMD called geographic atrophy patients. First 5 patients with visual acuity between 2100 and 2500. Next 7 will be between 2080 and 2500. We've started studies with the first couple of patients in the first cohort, and we hope to report the results by next year. So what I'm really interested in moving forward with is really a democratization of this autologous process to try to make autologous cell therapy available to as many patients possible. And one approach that we have taken is to collaborate with this company called Celino. based in Boston, which have used or is using a deep learning based approach to automate manufacturing of iPS cells. And the way this works is that the deep learning network recognizes iPS cells from non-iPS cells, and then they have a laser system, a very mild laser that comes and removes all non-iPS cells. Here you can see an example, recognizes iPS cells, non-iPS cells on the side, and laser kills all the non-iPS cells, and 24 hours later, so you have just iPS cell colony left, which of course can be expanded. And they're building a microfluidic setup, so the entire process is fully closed, fully automated, and we hope to have remove all the subjectivity from manufacturing of iPS cells and also be able to scale out to hundreds of patients simultaneously. At the same time, to disseminate all the knowledge and experience we gained from this work, we started an autologous iPS cell therapy consortium whose mission is to really to have a non-competitive space where we want to enable more autologous iPS cell therapies. And this consortium brings together experts from all various areas, academia, companies, nonprofit, really to exchange ideas, brainstorm problems, or broad interests across manufacturing, preclinical, regulatory, and clinical space, and really to enable more autologous cell therapies. So with that, I'll close. Thank the team that has helped get this done. And I want to also announce, thank the NIH for funding. All of this work was based off intramural funds from a big grant from Common Fund and additional funding from my own institute at the National Eye Institute. And thank you very much for your attention.
Thank you, Kapil. Before I jump in the presentation, I just want to say how exciting it is to see the data you just presented, Kapil, both as an ophthalmologist as well as the son of a loved one who has AMD, advanced AMD. It's just absolutely amazing. And so Amrita, I think your overall mission within MSCRF to sort of create a community together is actually being realized as I speak, because I'm going to follow up with you after this.
Absolutely.
Got a lot to talk about. Let's see, can everyone see this title slide here? Okay, terrific. So again, I wanted to thank Amrita and the MSCRF for inviting me. It's really an honor to be a part of this panel and also a part of a collaboration with the MSCRF, who have helped us both in advice as well as in financial support for some of our programs around RNA cell therapy, which is what the company that I lead focuses on, Cartesian Therapeutics. So I'm the president CEO of Cartesian. We are a clinical stage biotechnology company that was started in 2016 with the overall aim and a mission to unleash the full potential and reach of cell therapy through RNA engineering. And RNA engineering has several benefits over some alternative methods, and we see RNA cell therapy as the daughter of two parents that you're all probably familiar with. The first is what we call conventional RNA therapy. This is best exemplified by the RNA vaccines, where an RNA is put into a nanoparticle and injected in the body with the hope that the RNA is going to find its way to the relevant cells. Cells are going to take it up and express the protein that the RNA is going to encode. And our other parent is conventional cell therapy. Conventional in this sense, meaning cells being engineered with DNA, often introduced into the cell with a viral vector, and often these cells being immune cells. And so the typical example of these are the conventional CAR T therapies, where these cells are engineered and then outside the body and then introduced into the body, both personalized treatments that are autologous as well as off-the-shelf therapies that are allogeneic. So RNA cell therapy takes the best of both of these, if you will, RNA from the conventional RNA therapies and then the cell from the adoptive cell therapy world to introduce outside the body RNA into either an immune cell or a different type of cell. And currently we work with two different types of cells, T cells and mesenchymal stem cells. So some of the benefits in relation to conventional RNA therapy are the fact that you get minimal immunogenicity when you use the cell as the vehicle to introduce the RNA therapy into the body. You can therefore repeat dose within the patient. You can also engineer the cell to introduce proteins on the surface of it to allow it to home in on specific tissues. That's proven difficult to do with the conventional RNA therapies. And then with respect to conventional cell therapies with DNA, the primary benefit is safety because RNA has a beginning, a middle, and an end. It has a predictable half-life and there's no genomic integration. And as a result, the level of control you have over your therapy is at its highest relative to the conventional DNA-based therapies. You can also introduce multiple RNAs within a cell. So you can do true combination therapy. So you're not bumping up against cargo limits the way that you normally would with viral vectors and other mechanisms with DNA. And then, of course, finally, the reduced costs of using RNA can lead to significant benefits as well. So these are some of the benefits of RNA cell therapy. And historically, the RNA cell therapy has had some limitations that we've tried to overcome with some technology at Cartesian. So historically, large-scale manufacturing for repeat dosing has been difficult. Often these products have come out of single center studies, often in academic settings, where the degree of focus on the scale of manufacturing and the quality of the product has not necessarily been there. And also there really hasn't been a lot of work done historically on the length of RNA expression, which is obviously important to get a meaningful amount of efficacy. So we've developed a technology platform we've coined RNA Armory that is designed to overcome these limitations to generate a large-scale manufacturing with that allows repeat dosing with billions of cells per dose and yields a potent, high-quality product with extended RNA expression. And so this platform now has led to a robust pipeline with 3 clinical and 3 preclinical programs that are currently in development. The ones that are the furthest along are in our T-cell programs, and then you can see in the blue there, our off-the-shelf allogeneic programs are focused on the MSCs. And the one that I'll highlight here is the Descartes 30 program, which is being developed for patients with acute respiratory distress syndrome, as well as severe pneumonia, bacterial pneumonia. This is a program where we are taking umbilical cord mesenchymal stem cells and engineering them with 2 different proteins, enzymes, that are designed to work synergistically to degrade nets, or neutrophil extracellular traps. Nets are key drivers in the pathogenesis of autoimmune diseases, respiratory disease, and cardiovascular diseases. In essence, they are the neutrophils' attempt to control acute inflammation. And the way they do it is they undergo this programmed cell death called netosis, where they expulse their DNA that is studded with inflammatory proteins that serves as, in essence, a spider web to catch the viruses and bacteria that are causing the inflammation. The problem is they overdo it, and as a result, this net then serves as a nexus of inflammation in and of itself. And so with respiratory diseases, these nets plug up the alveoli, causing respiratory distress, as well as plug up the microvasculature, causing microthrombi. And so it stands to reason that if you can develop a treatment for reducing these nets, that you could treat the disease. And that's what we're testing out with Descartes 30 and these different indications. In order to be able to do this effectively, we decided to build the capabilities all in-house. So we don't really outsource much. So we have an integrated effort internally that allows us to be able to have full control over the quality as well as the manufacturing of the product here in-house. And it happens to be in the same setting as where we run our clinical operations and our R&D and discovery efforts. Our regulatory team is here, as well as our clinical enabling studies get conducted all under the same roof. And that level of integration has really helped us get products from preclinical development to clinical development quite quickly. So we're 5 for 5 in INDs over 5 years now. And we've developed a decent amount of regulatory know-how in the process and have gotten really familiar through over 150 manufacturing runs as to what works and what doesn't in terms of manufacturing these RNA cell therapies and these MSCs that are engineered with RNA. We've also along the way developed a cell bank for MSCs. So we bank umbilical cord MSCs that are GMP grade and clinic ready that we can reach into and engineer. And we're excited at the prospect of testing out some of these hypotheses in the clinic. And I look forward to the discussion that hopefully will follow in the next several minutes. So thank you. And with that, I'm going to pass it on to another colleague of mine that we collaborate with at Rooster Bio, John Rowley.
I really appreciate the invitation, Amrita and team. So I'm John Rowley. I'm the founder and chief product officer at Rooster Bio, and we're a very different company than everyone else's that's presented today, in the fact that we're not a therapeutic company. We enable therapeutic companies to get into therapeutic product development, scale up manufacturing on their path to commercialization. So just really quickly about Rooster Bio. Rooster Bio is about 50 employees. We were founded in 2013 right here in Frederick, Maryland. Our startup story was that me and my co-founder were sick of working at large companies and had an idea to really help to kickstart the regenerative medicine field by providing manufacturing friendly and off the shelf cellular raw materials and bioprocessed media, which would be incorporated in scalable manufacturing processes. We funded this by cashing in our kids' college fund. And we actually got our first products to market into our products into the hands of paying customers before taking any venture capital. And this allowed us to maintain a decent amount of control, proving that business model. And then we took venture capital to then to grow the business and to get our GMP raw materials on the market. So that's kind of our startup story that Amrita had asked that we share. And we've been growing this business now for almost 10 years. So what we do is we're really working on industrializing the regenerative medicine supply chain, and how we do that is that we believe there's a tremendous amount of redundancy in the field, where every therapeutic company that wants to incorporate MSCs within their product development efforts are required to establish their own cell banks and establish their own manufacturing processes. And what we're trying to do is standardize this by providing an off-the-shelf set of cell banks that are incorporated with off-the-shelf, easily implementable, scalable manufacturing, which we've developed ourselves. And so when we talk about industrializing the regenerative medicine supply chain, what we mean by that is that we designed our own GMP scalable MSC manufacturing process. We start from the tissue, whether it's bone marrow, whether it's cord, whether it's adipose. We then source those tissues under GMPs. We establish our own master cell banks. We have those master cell banks tested, which in every cell bank requires over $150,000 just in safety testing before you can start your clinical development, which is very, very difficult for many cell therapy companies to get into. From that master cell bank, we make then working cell banks. And from that working cell bank, we establish the processes for scalable manufacturing, whether it's in 10-layer cell flasks or even a large-scale single-use bioreactors. And then from within that context of the manufacturing process, we offer working cell banks, so that then any therapeutic company that wants to incorporate MSC technology into the products that they're developing, they can purchase off-the-shelf working cell banks from us, follow our production protocols to create then a very consistent final product, regardless from the many donors that they start with, and then do all of their product development and all of their GMP manufacturing using that same consistent standardized process. So that working cell bank we offer as a high volume MSC vial. So we offer vials of cells and bottles of media like many of the other suppliers, but they're all within the context of scalable manufacturing. And then we have GMP versions of all of our, well, not all of our products, but the majority of our products that people are taking into clinical testing, from the bioprocessed media to the cell banks, as well as then other technologies for generating extracellular vesicles from these cells, or for genetically modifying these cells. And so again, these are off the shelf. And what this does is it removes millions of dollars and years of time from a standard product development timeline. And we've helped some therapeutic companies get from product concept into approved IND in less than a year. So we do take a significant amount of time from this. We have focused and are proving our business model with HMSCs, these mesenchymal stem or stromal cells, because it's a foundational technology for tens of different therapeutic indications. Every year, there's over 150 MSC clinical trials started globally, ranging in indications ranging from regenerative medicine to immune modulation. There's engineered tissues and organs that are making it into the clinic. These cells can be genetically modified, as well as then these the secretome or the extracellular vesicles and exosomes can also be collected from these cells. So there's well over 100 companies that are and another 100 academic groups that are trying to take these into clinical development. And we are here to, again, standardize this very fragmented supply chain. Again, our goal is to radically simplify the incorporation of MSC technology into therapeutic product development by making them easy to establish within new programs. You don't have to have a 5 years experience in order to generate really high quality, high volume MSCs for product development. Once you start product development, we have manufacturing friendly product formats, these larger volumes, so anybody can very quickly generate enough for very large preclinical models, as well as for the manufacturing process development. And then we have off the shelf GMP versions of these products with master files that are on file with the FDA that anybody can reference. And again, so now the companies can focus on product development and less on that on the scale up manufacturing, which we have in place and offer to our customers. The basis for this is that as you scale your lot size from the laboratory scale of hundreds of millions into the early stage clinical trial billions of cell lot size to then the commercially relevant lot sizes of north of 100 billion cells, the cost per million MSCs will continue to come down, which will then get you to a commercially relevant both lot size and cost structure, so that you actually have a business at the end of the day. And as you scale up, these process development programs again they take years of time and millions of dollars, and as you scale up to hundreds of liters, these process development campaigns can get extremely expensive. And we've written blog posts on this, on how to establish multi-year, highly effective process development programs, helping to estimate what these lot sizes will be, as well as looking at the technology platforms which are required to be implemented. So please check out the Rooster Bio blog, for these, and it can be applied. We've written it around MSCs, but the technology and the concepts are applied to regardless of cell type. This is my last slide. Typically, from the standard, timelines for cell therapy product development can take 7 to 10 years, with a few years in research, trying to figure out what you want to do. Once you know the product you want to make, you have to get into manufacturing process development, and then redo many of your experiments, based off the cells coming out of that GMP manufacturing process, before you can then tech transfer and get into clinical manufacturing. And by really offering these off-the-shelf, highly relevant cells with manufacturing built in, you can quadruple the pace of research. You can really speed through product and process development, and with then off-the-shelf master cell banks that take 2 to 3 years to establish, you can really get that product manufactured and again get into the clinic in only a couple of years. And so we believe that this model is going to be and it is already starting to be applied to other areas of the regenerative medicine supply chain. And I believe that someday we will be like technology products, where you can get off-the-shelf transistors and off-the-shelf microchips and off-the-shelf screens and GPS, and really make these convergent technologies very rapidly get into clinical development and testing. And then once something works and we know what works, speeding through clinical development and getting these products into the clinicians' hands to be helping patients, with these unmet medical needs. So with that, I'll stop and get on to the panel. So again, thanks again for the invitation.
Great, thank you all so very much. And, I feel fortunate that I've had several conversations with each of you, and now is the hard part of trying to, fit technology, all of your experience and everything into our short panel discussion. But yes,
I forgot to mention that, during us developing our products, we got 2 MSCRF product development grants. And we launched 2 products, one a large-scale cell bank, as well as one, product for enhancing extracellular vesicle production from MSCs. And we launched those out in the world. So again, we've definitely benefited from having MSCRF within this great state of Maryland.
yeah, thank you. And we've definitely benefited from having all of you in the community and giving back. And maybe that's a point I'll touch on later, because all of you almost highlighted so many of these collaborations you have ongoing, which is so critical. But perhaps I'll first start with the audience. Obviously, several of you work with, have been, or still are scientists, physicians. And so maybe we can start by talking about key considerations that you think the audience should take away, because we've talked about different cell types, different technologies, different indications. So what are sort of key differences between developing products and therapies based on pluripotent versus non-pluripotent starting material? Perhaps if you want to comment on that.
So, I think you can, I can start with MSCs, right? We're all about efficiency, and we've been thinking scale up and efficient manufacturing and the impact on cost of goods for a very, very long time. We have a manufacturing process that goes from working cell bank to billions of cells, and it can go to hundreds of billions of cells, all within the context of about a 8 to 10 day manufacturing process. Right. So you can run, a good, 3 or 4 of those manufacturing processes a month, within the context of a large scale GMP manufacturing facility, once these products actually get to market, right? And so it is really important to think about, the speed of manufacturing, the scale of manufacturing. And as you get to hundreds of billions of cells, that lot size is still only between 500 and say 2,000 product doses per manufacturing lot. And if you're trying to treat hundreds of thousands of patients a year, then that lot size is probably going to have to go tenfold above that and really get to that trillion cell lot size, which is kind of scary. but it fits within the context of today's single-use manufacturing products, at least for MSCs. Very, very different, for than some of the autologous, production programs. But then again, at the same time, there's definitely a time and a place for autologous, and the patient populations aren't necessarily. It's not like they're going after stroke, right? Which is which there's hundreds of thousands of patients every year, and there's definitely indications where autologous is very, very appropriate.
And if I can add there, so I think the big difference is, as John pointed out, is between iPSC or pluripotent versus non-pluripotent is the manufacturing process is significantly longer. But for that reason, most of the diseases indication that the pluripotent derived cell therapies are targeting are slow progressing degenerative diseases. So there's this time to manufacture. In terms of manufacturing, because unlike MSCs, which we have been using for a long time in clinical practice in various different ways, there is still not optimized ways to manufacture iPS cells, optimized as in clinical grade, regulatory approved, streamlined path forward. So a lot of us are trying different ways of reprogramming, different ways of expansion. And I think sooner or later, there will be a convergence towards, what are the most approved, most feasible ways to manufacture. And for that reason, because it's a reprogramming process involved, there's this concern about safety coming, or safety of the cells or derived products. So the regulatory oversight on this has to be major, especially for GLP tox studies, at least at this stage. And that's what all the groups are focusing on. In terms of moving forward, as you heard from Doug, I think, and from my work, a lot of these things all of us are doing are still proof of concept studies. But this may not be the actual final path we all go forward with. Either I think this may change with either, hyperimmune platforms that Doug pointed out, or automated manufacturing. So various versions of this will keep changing with time. But I think pluripotent stem cells, they provide, for me, a different venue. They are pluripotent derived cell therapies provide a replacement tissue, right? For instance, in our case, RP cells, or in the case of Doug, part of muscle cell through stem cell population. And that's the key difference between the two technologies. And that's why I think both of them have an essential path going forward in parallel to patients.
That was really great, Kabir. I'll add a few different points that you kind of alluded to too, just to put some more color onto it. So you were referring to the regulatory component, right? And I think that is one of the key differences between developing, an iPSC derived technology versus a non, iPSC derived technology, right? And at Vita, we have both, right? So we're kind of in this dual strategy where we're currently focusing on both. And I think one of those challenges with iPSCs is that because it's a relatively new approach from commercialization, right? I think the first IND was accepted back in 2018. The FDA puts a lot of emphasis on safety, right? So I know Kapil and I, we've spoken about this in depth, but the FDA now guides about a 9 to 12 month safety study where you inject your cells into an in vivo model, observe them for 9 to 12 months and ensure that they're still in that differentiated final cell type, right? It's really important for them to know that they're not, continuously going to be pluripotent and mutate into anything else, right? So I think that's one of the key differences. I look at our immunotherapy platform. Our safety study is really about 12 weeks, right? So that extra 6 to 9 months, it adds a substantial amount of time and cost. And unfortunately also adds, risk, right? Things that can also, go wrong as you look to start that study, right? So I think that's one key difference from the regulatory component. And then on the first points that you were making, Kapil, the challenges with iPSC is that, there isn't really a streamlined manufacturing process, right? And we're all kind of working with a bunch of different technologies. And as you talked about, there's a bunch of different reprogramming technologies out there as well. But one thing that I just learned that I think is really helpful, and I'm really glad that this is the shape that the iPSC space is taking. But it sounds like the FDA and some of the regulatory agencies are going to be willing to accept iPSCs as a starting source. So what does that mean? Well, it means that, right now we're working with a specific CDMO, and maybe in the future, it makes sense for us to switch to a different reprogramming protocol, right? Maybe you want to switch from Sendai to, and like an mRNA based, technology, right? Or it looks like the FDA is now going to allow you to kind of use that starting source as the starting point for each individual iPSC-based therapy. And all you would have to do is do a comparability study on the two different starting sources. So I think that's really huge, and that's moving in the right direction. And I think that'll help streamline manufacturing a little bit, because it's not going to tie, individual programs like what we're both doing into a specific technology. It's going to allow us to make better, decisions as a lot of these different manufacturing, processes get proven out in the clinic. So I'll stop there, but.
Yeah, and Murad, if you want to continue along those sort of regulatory discussions, because clearly you've had a lot of success in this, you want to comment on the process and how you've navigated it?
Yeah, so, I think we're fortunate that we work with RNA, where I think, and we're fortunate we work with RNA at a time in 2022 when you have a couple of RNA therapies and RNA vaccines that were approved based on a wealth of just terrific clinical data. So, I think, the fact that we are reprogramming our cells, but doing it only temporarily, has a lot of benefit from a safety perspective, and as a result from a regulatory perspective. The notion that we would be tinkering with our cells by permanently modifying them, irreversibly modifying them with DNA, and then expecting us to have gone from preclinical to clinical with as many programs as we have would be unthinkable. So I think just RNA engineering has, just by its very nature, that regulatory advantage over sort of the conventional way to engineer cells with DNA. Now, the Kalpita and Doug have added an additional layer of consideration that I actually wasn't aware of. And of course, it makes a lot of sense. The iPSC is requiring a very extended safety package from a preclinical perspective. Makes all the sense in the world for the reasons that they pointed out. I wonder, and I mean, this is, I guess, a question to Kalpita and Doug. Are you all aware of successful early stage clinical programs that deploy RNA engineering into iPSCs?
I don't think I know of any program that has used RNA engineering for reprogramming or RNA-based reprogramming and has gotten to an IND stage. I know a few that are in the process that are doing IND enabling studies, but I don't think those INDs have been submitted yet.
Yeah. No, I can't think of any that actually got the INDs accepted either. But there's two that come to mind that I didn't even know they had moved to IND enabling studies just yet. But they're working on that approach from a reprogramming standpoint.
All of you spoke a little bit about your commercialization process, and thank you for sharing your startup journey story as well, because I think it's really useful for those starting out on this path to hear it, because all of you have had distinct paths and have been hugely successful at it. But are there any other key considerations you think someone starting out on this path should think about a process of commercialization and how one goes about it? We've sort of touched on regulatory. You've mentioned in the passing of deciding whether to work with CDMOs or building your own manufacturing, and that's a decision. And there was some alluding to like the QC and the testing required. But I just want to pose a broader question to see if there's anything else you want to discuss or dive into details that we might give people to think about as they think about commercializing their technologies.
If I may start on that, and I know John and Murad and Doug probably have much more experience than I do. I'm still in academic space, and I'm only licensing out our technology to commercial partners. I think, especially with these cell therapies, and more we're trying to develop complex cell therapies. replacement tissues from iPS and ES and different versions of iPS. These are complex things. These are living tissues. And I think they require a lot of know-how, a lot of fiddling around at early stages. So one key advice I can give is that if you license out a product to a company or start a company, stay with it as long as you know the product is ready. Don't just say, I handed it out and I'm done, I'm going to move on and do something else. As myself who's doing it now, I'm working with 2 to 3 different companies who have different parts of our technology. And I see there's a lot of hand holding required to make sure that every key aspect of the manufacturing process has been properly transferred. And as we're doing our phase 1, we are further optimizing the manufacturing process. We are further improving the reagents that go into it. And that's what the part of a phase 1 trial is. So the only thing I would tell to academic scientists is just stay with your technology. You need a lot of patience and don't just give up at early stages. Getting to phase 1 is just the start of the journey. That's not the end of the journey. We have a long way to go from there.
I mean, I would echo that in the strongest terms. There is a world of difference in optimizing a technology at a sort of research scale versus a clinical scale. The cells behave differently when you scale them up. We've seen this time and time again with these reprogrammed cells, the RNA cell therapies that we work with. So the level of risk you take on going from a preclinical scale to a clinical scale and then from preclinical stage to clinical stage is astronomical. And so to the degree that and there's so many nuances in the tech transfer that's happening. If you're in an academic setting and you're working, as company said, with a commercial partner, those nuances really do matter. And that iterative optimization as you're scaling up really does matter. And so it is not as simple as a handoff. And so it's not as easy as publishing a sexy paper in a highly esteemed journal, and then taking that and taking the IP associated with it and licensing it out and expecting it's going to go well. It's just, doesn't happen. It doesn't happen often. And so when you choose that kind of development partner, I think you want to choose someone that has that background and experience in executing and a real eye towards that iterative optimization, towards taking something that is sort of a research grade scale to a clinical scale. Once you start to generate high quality product at clinical scale and start to get some interesting proof of concept data in the clinic, things start to happen on their own, and they pick up speed. As Kapil said, that's still only in the early stages. Ultimately, I think people are probably familiar with these statistics. From the percentage of assets that go from getting into the clinic and starting phase 1 into going all the way and being marketed under a BLA are about 15%. So 15% of the assets and only about 6% of their oncology assets. So it is enormous risk all over the place. It's sort of crazy to think about, but you want to get it right when you choose the right partner in the beginning.
Yeah, and I would say, for technologists, which I think is the main audience today, do not underestimate the, the operational and business aspects that it takes to actually get something out of a university and into a company. I think most of the business people here are technologists at heart and understand what it takes to do that. But the technology is really the core technology that comes from a university is I would say 15 to 20% of the work, and then the rest of it is the amount it takes to, just to the infrastructure to have a company running, to do the finances, to pay the rent, to keep all the legal and the intellectual property in place. It does take a village to correctly commercialize a product. And I don't even have clinical operations, right? I mean, the stuff that the therapeutic companies have, are orders of magnitude more complex on the clinical and regulatory operation side. We still have regulatory because we do have our things in master files with the FDA, but we're not getting anything approved, for therapeutic production. And we might have a more complex, supply chain, and because we're constantly shipping things to customers and getting orders and a little bit more financial, but everything is hard. Every little piece, even shipping, is challenging, and definitely do not underestimate the business and the operational aspects of building a business.
Yep. Doug, do you have anything to add? I know
mean,
you work very closely.
John's hitting on some key points and some also like some painful points that we've been through. So that's why I'm smiling over here, because I know exactly what you're talking about. And just to maybe add some color to what John's saying there. So, we specifically when it comes to our satellite cells VTA100 program, we licensed this out of Hopkins. And at the time, it was very much a research protocol. And I would say maybe 80% of that protocol had components that you could never even use from a commercialization standpoint, right? So as we licensed that out, we were doing, the tech transfer from Hopkins into Vita's lab. We were working on optimizing that protocol in itself, just from a research standpoint, and then started working with the CDMO where we're doing a second tech transfer and trying to optimize that to a GMP protocol. So it's just several layers of complications. And, we look back now, and I can smile and take a deep breath now because we've basically been through the heart of it. But it took about 2 years for us to go through that process to get from that almost completed research process to now that GMP validated protocol, right? And it's no easy feat, and especially going from a tech transfer of working with other folks. Right. So certainly, even the shipping, that's why I was smiling, because we had a few delays over the years where, you're working with a specific shipping company and they didn't store the cells at the right temperature. And then all of a sudden, you lose something. Right. So those little things, they certainly happen on a regular basis, but it's it's a complicated process when it's all said and done.
No, thank you. And it just gives me great pleasure to hear you all speak, because part of this is you exemplify this process that we have, moving research through validation into companies, and as well as some of your clinical trials. Beyond that, also the fact of how much building an ecosystem has been key to our success, and you're all such a huge part of this. And so almost all of you refer to it in your presentations, but I wonder if you'd want to comment a little further on the importance of collaborations to advance science in this industry, and what ongoing collaborations you have and how people can work with you or what you're looking for.
If I may start again, I just want to actually even go back to the previous question. And I want to say that, the goal of this is, I know a lot of us pointed out complexities of the cell therapy program, but the goal is not to discourage anyone. The goal is to set the expectations that it requires hard work and patience. But I want to say that upfront, the most important thing for any academic scientist who's going to be attending ISSCR is robustness of science. If your science is robust, you make a product that is really high quality, and you're hearing from Doug, tech transfer from lab, from university to the company to CDMO. If you do not cut corners at every stage, you will find the right partners who will take you to the next level. So I just want to make that, as a positive closing of that question. In terms of collaborations, I think, again, it's finding the right partners who have the same belief that you have in your science, in your product, and can help you, raise funds, do logistics. You have all the experts here on this panel who can tell you a lot more. Working on the next generation technologies, like, we're working with this company, Celino, that I hope, if the technology works using AI and microfluidics and laser that they have, we can fully automate the entire manufacturing process. And as you're hearing from Doug, if the FDA takes now iPS cells as a starting material, then that simplifies the entire manufacturing process several orders of magnitude. So really, I think looking for those kind of collaborations that where you can build upon your initial technology and find partners who can, first of all, enable that first technology and then build upon there to develop more futuristic technologies is what one needs.
I can go next. I mean, I think that every company has their strengths, right? And but in order to have a complete solution, you just need to partner with complementary companies, right? And so we provide cell and media products that are associated with large scale manufacturing, but we don't make any production equipment. And so we have actually a long history of collaborating with these bioprocess providers that have bioreactors, that have new cell culture tools, and then demonstrating, I guess, scalability within these within different bioreactors that different institutions have, whether it's hollow fiber bioreactors, stirred tank bioreactors, vertical wheel bioreactors, and establishing the protocols, right? So it's easy for us to get access to cells and media, which is usually the largest chunk of a bill of materials of any product. And so these equipment providers, it's very expensive for them to establish these protocols, and it benefits both parties to have an established MSC protocol for a specific piece of equipment. And so we've been very successful in establishing collaborations, again, with these different bioprocess equipment, whether it's bioreactors, whether it's downstream processing equipment, and then you get publications out of it. You get white papers and posters to present at conferences. And then what we find is that when our customers are looking for rapid ways to get their GMP products, they figure out what equipment their institutions have in their GMP, whether it's a quantum bioreactor or a stirred tank or just 10 layers, and they work with us. Well, what's the best way to do this? And we can easily coach them on that path, because really time is money when it comes to getting these things into the clinic. So
I'll say, John, I think starting with a number of different collaborations can really help you move a little faster out of the gate, right? But I think the goal should be, long term, to always try to work towards having kind of a combination of the two, right? And Murat, you've done a fantastic job of really setting up, an in-house process for your GMP work, and even sounds like your IND enabling studies. And I think I look at a lot of what you've done and say that those are some goals that we have here at Vita, but I don't think we could have started there, right? We have 3 different GMP projects ongoing. We chose 3 different CDMOs who we felt really comfortable with for those specific cell types, right? One day, if we can get to a point financially where we can make the decision and start doing it in-house, I think that's going to be very beneficial, because it can help us, reduce cost in the long run. But helping and partnering with those partners really allowed us to kind of move, quickly and swiftly into that preclinical process, and hopefully into the IND enabling process in the not too distant future for all 3 of those programs. So I think it is really important to kind of find the right partnerships really early on, but then also just start thinking about ways, to be a sustainable, therapeutic company or biotech company, to bring in some of those partnerships at one point in-house as well.
So yeah, no, that's I echo that. When we first started, by the way, we didn't have everything in-house, nor were we planning on having everything in-house. So, my previous company, we started with 2 people at the discovery stage and ended it at the registration stage, the phase 2b/3 study stage, with 2 people. So we outsourced everything. And when we started Cartesian, a week after we sold that previous company, we were thinking of the same mindset. We were just going to do it all virtually and focus on, these RNA cell therapies, but just outsource everything. And we started doing that. We even contracted with a GMP manufacturer, who shall remain nameless, that we realized just wasn't going to work. Not that they didn't mean to do a good job, but because unlike small molecular development and unlike sort of antibodies and other biologics, working with cells is very difficult. Cell therapies is sort of the Wild West from both a technology perspective as well as a regulatory perspective. So we wanted to have control, and we realized we had to have control over the entirety of the process, which is why we made that investment and built the facility here and just organized our entire company around that GMP manufacturing facility. Now, that doesn't mean we don't collaborate. We certainly do. And even within Maryland, within the world of MSCRF, we work with John and his group at Rooster Bio. And I think everybody who works with MSCs is either has or eventually will cross paths with John and Rooster Bio, because they do a terrific job and they're very easy to partner and work with. We also work with our clinical operations group with a whole bunch of different institutions, but in particular, the University of Maryland and their critical care unit, where the proximity allows us to be able to get fresh samples from patients and be able to analyze them on the same day, which affords a lot of terrific data that otherwise wouldn't be possible with these types of cell therapies. And then we license technology from the NIH as well. So, ultimately, you can't really get anywhere without these types of collaborations. I fully agree with you. And I think they're particularly important in the world of cell therapy when the paths to take a drug from discovery to commercial are not as well delineated as the traditional drugs that we're all familiar with, small molecules and biologics.
No, absolutely. And I mean, Doug also mentioned different work he has ongoing with Hopkins. So I know all of you are hugely involved in this community and sort of helping us minimize that gap between academics and industry to help move these out faster. But Couple makes a good point. I feel like this field is moving so fast that we're always talking about the next thing and the challenges, which is why it was great hearing some of your stories on how far we've actually come. And even when I look back, since we put in place these accelerating cure initiatives, we've supported over 20 companies, and all of you have raised between 6 and 40 million at least in follow-on funding. So, we've really come a long way. So the point isn't to discourage anyone, it's just to take everything under consideration. But maybe in that spirit, I should ask you, what are you most excited about now? And where do you sort of see the industry or your organization in the next 5 to 10 years?
I'm happy to just start there. I mean, I think, we're probably all biased. I think we're probably all thinking the same thing, but I think cell therapy it's never been more exciting to be in the cell therapy space. I mean, I think about just from a pure investment perspective, right? If you go back a few years ago, the technologies that we're working on here at Vida, nobody was getting funding for them, right? I mean, as it relates to VT100, we're combining gene editing with iPSCs, and we have an extensive differentiation protocol. I mean, it's pretty complicated stuff, and here we are, able to attract the funding and potentially deliver a cell type that can really be restorative to patients instead of just potentially trying to diminish their symptoms, right? So I think cell therapy as a whole has never been more attractive, because I think there's a real appetite for it, as it exists from an investment standpoint, and then the infrastructure is growing too. Right. Probably one of the, earliest challenges, which is, still a challenge to an extent, was that there weren't a lot of CDMOs out there, and there weren't a lot of CROs and people who knew how to advance this type of technology. But that's growing. Every day or every month or so, I feel like I hear about another, just an iPSC, startup CDMO who's kind of going out there and trying to move this forward. So I think it's never been more exciting to kind of be in this space. And in the next 5 years, I think this field's going to grow tenfold, and hopefully we're going to see some really impressive clinical results that support that and allow us to kind of keep growing.
I'm really excited to see more and more of these off-the-shelf components, being offered, whether it's lentivirus, whether it's iPSC cell banks, whether it's MSC technology, as well as other things, be offered that really will help to standardize the field and accelerate product development. So we're gonna go from having things take several years to just a couple of years, and eventually just take a few months to go from concept into the clinic. And I think that that's going to just be a really exciting time. And I really do believe that biotechnology will be paralleling the progress that the technology field has had. We're just a little bit farther behind because we knew how to process silica before we could knew how to process DNA in cells.
I can go next and leave it to you couple to close it out. But I mean, from my perspective, cell therapy takes advantage of millions of years of evolution. And we are not smarter than evolution in this perspective. We are, in essence, harnessing that learning over that length of time, and our ability now to reprogram cells that allows us to use the cell both as a factory for producing as well as a vehicle for delivering not just one therapeutic, but multiple therapeutics right to the site of disease, I think is going to change the world. Having said that, there are heightened expectations for cell therapy that may be exceeding what the field as a whole can deliver over the next 5 years. Maybe not 10 years, but perhaps the next 5 years. And so, I think the innovation tends to happen incrementally. And I think if you sort of think of where we are at as a field for cell therapy and stem cell therapy in particular in 2022, I think in 5 years, it's entirely feasible and reasonable for us to be able to use the power of the cell to deliver a combination therapy for a slew of diseases, and perhaps even get a couple of these therapies approved. And upon approvals, upon the first couple of such approvals, I think that'll open the floodgates that will allow the kind of innovation to accelerate to the point where in 10 years, we've changed the world with cell therapy.
I think really there's not much more to say. Murat and John, you said it all. Really, I think you're absolutely right. What we need, what the MSC or non-pluripotent, pluripotent stem cell therapy really need is something what happened to CAR T and gene therapy, right? A couple of approvals, and then really the floodgates will open. But there is now path to get there. I think, as Doug pointed out and Don said, there's tools available. There's venture interest in there, all the key things that we need to put together to make it happen. And that's what we're going to see in the next 5 years is more and more people putting together their ideas and saying, let's get to the clinic, let's get to the first patient, let's get to first in human trial. And then as some of the other work goes forward, we will see much more progress in this field. I think already in the past 2 years, I saw many more autologous IND being started, enabling work or GLP works being started. And we're going to see many of those reach and complete first in human trials in the next 5 years. And really, the important point, I think Murat just touched on it, is we also have to be mindful of that the expectations have to be set right. Like anything, any other drug, this is yet another drug. There's no magic bullet in it. Stem cell therapies may work in some cases, may fail in other cases. We'll have to go back to the drawing board and we'll have to redraw, we'll have to change our products, and we'll come back and come back with better therapies. So that this is just for lay audience that sometimes comes to ISSCR, that there is no magic in stem cell based therapies. This is just like any other drug. It's an iterative process, but there's a lot more enthusiasm and right factors are there to make them happen. And I think it will happen more and more in the next few years.
Well, thank you. Thank you all for your time and your insight. It's always fun talking to all of you.